eminent speaker, Professor B.S. Kaith, Head Department of Chemistry, NIT Jalandhar. And fourth, respected eminent speaker, Professor S.K. Tripathi, Department of Physics, Punjab University, Chandigarh, organizing secretary of this webinar, Dr. I.D. Sharma, Assistant Professor, Department of Physics of our college, convener, Shri Bhupendra Singh, Assistant Professor, Department of Physics of our college, distinguished participants, guests, dear friends, ladies and gentlemen. First of all, I welcome you all in this national webinar on recent advances in material sciences and nanotechnology, organized by our physics department by depth of my heart, from every nook and corner of my heart, rather I can say. It is our privilege that today we have conjunction or confluence of eminent scientists from different parts of India, from Bengaluru, from Kanpur, from Jalandhar, Chandigarh, Allahabad, Meerut, and many parts of our country. And the credit for this goes to especially Dr. I.D. Sharma, who has taken pains to organize this webinar, and he has invited a large number of dignitaries in this function. He deserves wide appreciation, and I congratulate him for organizing this seminar. Although Shri Bhupendra Singh, another our colleague in physics of department, he also deserves wide appreciation. The topic of this webinar, that is recent advances in material sciences and nanotechnology. This is very fast moving, I can say, research area nowadays. As I am a student of biology, a student of botany, but I have read physics up to 12th standard. And I can say that material sciences and nanotechnology, material sciences, that is design and discovery of the materials, especially solids. And nanomaterials, that is small sized materials which have at least one dimension less than 1 to 100 nanometer. And this, the use of these nanomaterials, that is nanotechnology. This is nanotechnology in broad sense. And this nanotechnology or these nanomaterials, these are becoming commercialized nowadays. And there is these nanomaterials, these are present naturally, as well as these can be produced by different by combustion processes, or they can be engineered. And these nanomaterials are being used for different manufacturing processes, for example, paints, insulations, lubricant additives, and also in healthcare units. Nanoenzymes, that is a common term. That is many nanomaterials. These act as enzymes. They have enzymatic activities, and they are being used in health sciences or healthcare. Similarly, drug delivery, that is transportation of the different chemotherapeutic drugs. These can be transported to the site of cancer. This is another important use of these nanomaterials in healthcare unit. Similarly, in cardiovascular diseases, these are being used. And in controlling water pollution, these nanomaterials, so this nanotechnology, that is also being used. There are many experts. There are 4 eminent speakers who will speak on this topic in this webinar. So I have said brief thing which I know in broad sense about nanotechnology. Again, I hope that this webinar will be successful in achieving its goal, and the participants will be benefited to a great extent. At last, I once again I take this opportunity to again felicitate and welcome all our guests, participants in this webinar. Thank you very much. Jai Hind, Jai Bharat.
Thank you, sir. Now I greet and welcome to our guest of honor, Professor Rajiv Gupta ji, Regional Higher Education Officer, Meerut. As I come to know that he had done his MCom, PhD and LLB from Agra University, Agra, MBA from Indira Gandhi Open University, Delhi, and MJMC from UPRTOU. His broad area of interest is commerce and business administration. He is a good academician and good administrator. He had 21 years of teaching experience of undergraduate, postgraduate students classes and 2-year industrial experience as senior manager finance and corporate management auditor. He has supervised 90 research projects and his 15 research papers are published in highly impacted reputed journal. Now over to Professor Rajiv Gupta ji, please.
नमस्कार सर, मेरी आवाज आप तक आ रही है?
नमस्कार, नमस्कार सर, नमस्कार।
नमस्कार सर।
आ रही है गुप्ता जी, आ रही है।
एक तो मैं सबसे पहले मैं महाविद्यालय में आज आयोजित एक दिवसीय वेबिनार रिसेंट एडवांसेस इन मटेरियल साइंसेज एंड नैनो टेक्नोलॉजी पर आयोजित इस कार्यक्रम में उपस्थित महाविद्यालय के सहयोगी प्राचार्य डॉक्टर नरेश मलिक जी और हमारे जो एमिनेंट स्पीकर हैं एम ऐश्वर्या मूर्ति, डॉक्टर सिद्धार्थ पांडा, बीएस कैथ, एस के त्रिपाठी जी और ऑर्गेनाइज सेक्रेटरी डॉक्टर आईडी शर्मा जी, कन्वीनर श्री भूपेंद्र सिंह जी और विभिन्न मेरे प्राध्यापक साथियों और छात्राओं और हमारे जैसे कि आज बताया था सर ने कि हमारे मुख्य अतिथि जो आज हमारे निदेशक के रूप में उन्होंने प्रभार भी लिया है, संयुक्त सचिव डॉक्टर अमित भारद्वाज जी, वो नहीं जुड़ पाए किसी कारण से। मैं आप सभी का अपने विभाग की ओर से तहे दिल से स्वागत करता हूं। ये मेरे लिए बड़ा सौभाग्य का विषय है कि मैं आज इस वेबिनार में अपने विचार साझा करने के लिए आपके साथ जुड़ सका हूं। पिछली बार भी इस महाविद्यालय के द्वारा न्यू एजुकेशन पॉलिसी पर वेबिनार आयोजित की गई थी, परंतु दुर्भाग्यवश कुछ संचार व्यवस्था के कारण हम लोग साझा न कर पाए। आज जो विषय भौतिक विज्ञान संकाय द्वारा जो चयन किया गया है, बहुत ही समीचीन विषय है, रिसेंट एडवांसेस इन मटेरियल साइंसेज एंड नैनो टेक्नोलॉजी। इस विषय पर ही पूरी 21वीं सदी आधारित है और वास्तव में जो हमारा जीवन है वो जीवन इस बात में उसकी उपादेयता नहीं है कि हम अपना कितना लंबा जीवन जिया। उपादेयता यह है कि हमारा सूक्ष्म जीवन हो पर महत्वपूर्ण हो। यही नैनो टेक्नोलॉजी का सार है, यानी कि हमारे पदार्थ सूक्ष्म हों और हल्के हों, यानी कि उनका उपयोग अच्छा खासा हो। 1 मीटर के 1 अरबवें हिस्से के द्वारा जो पदार्थ बनाए जा रहे हैं उनका उपयोग हमारे विभिन्न क्षेत्रों में किया जा रहा है। जैसे कि आज हमारे विभिन्न शरीर के हिस्सों में अंदर जाकर के कैमरे के द्वारा जांच करनी, ये नैनो टेक्नोलॉजी के माध्यम से ही संभव हो सका। हमारे जितने भी कार्य हैं वो कार्य जब ही संभव होते हैं जब हम सूक्ष्म स्तर पर उनको सफल करते हैं, और सूक्ष्म स्तर पर कार्य होते होते ही वो बड़ा रूप धारण कर लेते हैं। आज हमारे जितने भी महाविद्यालय हैं उनमें सबसे ज्यादा आवश्यकता ये है कि हम शोध कार्यों पर हम बल दें, और जब तक शोध कार्यों पर बल नहीं दिया जाएगा हमारे महाविद्यालयों की उपादेयता नहीं रहेगी। मटेरियल साइंसेज एंड नैनो टेक्नोलॉजी, ये भौतिक विज्ञान के अंतर्गत विद्यार्थी ये अध्ययन करते हैं, और निश्चित तौर पर ये नैनो टेक्नोलॉजी के माध्यम से ही आज हम विभिन्न क्षेत्रों में विकास की ओर अग्रसर हो रहे हैं। जिस आज हम देखते हैं कि जब हम अपने फार्मिंग के लिए जब हम पानी डालते हैं पेड़ों पर तो काफी पानी हमारा व्यर्थ हो जाता है, उतना पानी आवश्यकता नहीं होती। जबकि यदि हम केवल उतना ही पानी दें पेड़ों को जितना वास्तव में उनकी जरूरत है, तो स्प्रे के माध्यम से तो हम पाएंगे वो पानी हमारा व्यर्थ जा रहा है, उसका उतना उपयोग नहीं है, और उस पानी को हम कहीं और प्रयोग कर सकते हैं। ठीक इसी प्रकार हमारे बहुत से ऐसे क्षेत्र हैं जहां हम नैनो टेक्नोलॉजी के प्रयोग के माध्यम से हम अब हमारे पास जितने भी पदार्थ हैं उनका हम बेहतर उपयोग करना संभव हो पाएगा। हमारे विद्यार्थी जो आज विज्ञान पढ़ रहे हैं उनके लिए नैनो टेक्नोलॉजी के अंतर्गत रोजगार के तमाम अवसर उपलब्ध हैं। हमारे विद्यार्थी विभिन्न प्रकार के जितने भी प्रोडक्ट्स हैं उनकी डिजाइन किस प्रकार हमारी सूक्ष्म सूक्ष्म हो जिससे उनकी लागत कम आए। जैसे कि आपने देखा कि जो टाटा कंपनी द्वारा नैनो कार लाई गई उसका भी एकमात्र यही उद्देश्य था कि नैनो टेक्नोलॉजी के माध्यम से हम कम लागत पर उस पदार्थ को लेके आए। निश्चित तौर पर उसकी लागत कम थी, परंतु लागत के साथ साथ एक और जो विडंबना रही कि एक और हमारे यहाँ एक विचार होता है कि जिसकी जितनी कीमत कम होती है उसकी उतनी कीमत उसका मूल्य उतना कम आका जाता है। क्योंकि सस्ती थी तो इसलिए उसकी वैल्यू उतनी नहीं मिली। हालांकि चाहे वो महंगी कार हैं चाहे वो सस्ती कार हैं, वो कार्य वही कर रही हैं। हमारा भी उद्देश्य महाविद्यालय में होना चाहिए, हमसे कम लागत पर अधिक से अधिक हम किस प्रकार हम उत्पादन कर सकें, किस प्रकार अधिक से अधिक उनका उपयोग हो सके। महाविद्यालय में भी हम सभी सारे विद्यार्थी प्राध्यापकगण प्रयास ये करेंगे कि इसी नैनो टेक्नोलॉजी का जो बेस है इसी को अपना करके हम अपने महाविद्यालयों में शोध कार्य को बढ़ावा देंगे। मैं इस विषय का विशेषज्ञ नहीं हूं, मेरा इसमें रुझान है, कवि नहीं हूं किंतु करता हूं काव्य से प्यार हूं, और जिंदगी असली कहानी मैं कहानीकार हूं। मैं केवल इसमें बहुत रुचि रखता हूं, और मेरा आज का जो संबोधन है मैं बहुत उम्दा नहीं बात रख पाऊंगा क्योंकि ज्यादा इसमें बहुत ज्यादा एक्सपर्टाइज नहीं है। और एक और अभी वेबिनार है हमारे शासन की, वहाँ मेरी वहाँ व्यस्तता है। मैं साधुवाद देता हूँ महाविद्यालय परिवार को और हमारे आईडी शर्मा जी को कि निश्चित तौर पर साधुवाद के पात्र हैं। उन्होंने मुझे अवसर दिया कि मैं अपने विचार साझा कर सकूं, और निश्चित तौर पर ये वेबिनार सफल होगी और इससे लाभान्वित होंगे सभी लोग। बस मैं इतना ही कहूँगा कि हम अपने प्रयास जारी रखेंगे और निश्चित तौर पर एक ना एक दिन हम कामयाब होंगे। जय हिंद, जय भारत, धन्यवाद।
Thank you, sir, for your insightful talk and kind Bliji. Thanks once again. Thank you, sir. Professor Murthy. Now, I welcome to our eminent speaker, Professor M. Murthy ji, from Jawaharlal Nehru Center for Advanced and Scientific Research. I welcome and invite Professor M. Iswarmurthy. Professor M. Iswarmurthy obtained his PhD in 1906 in chemistry from Anna University under the supervision of Dr. Professor B. Krishnan Swamy. Then he has spent 2 years with Professor C. N. Rao, Fellow Royal Society, as a postdoctoral fellow at JNCSR Bangalore. Later, he moved to Japan under AIST Fellowship and worked with Professor Fuji Mizukami, former director AIST Sendai, for 3 years. He has also spent 2 years as a postdoctoral fellow in Bristol University with Professor Stephen Mann, Fellow Royal Society, United Kingdom, before joining as faculty at JNCSR in October 2004. His research interests include nanoporous material for catalysis and adsorption, hybrid material for drug delivery, and energy harvesting. He has more than 150 research papers of highly impacted journal. He got MRSI medal in 2011, Material Research Society of India, and CRSI bronze medal in 2015, Chemical Research Society of India, and CNR Rao National Prize for Chemical Research 2018. Now, I welcome and invite to Professor M. Iswarmurthy for his talk on nanomaterials for biological application.
Thank you, Professor
Thank
Sharma,
you.
for the nice introduction. Can you hear me?
Yeah, sir.
Okay. Yeah. Thank you so much. I'm really happy to be here in the part of this webinar, and it's also so happy to see this PG college where that Sanjeevi Choturam College, who I understand who's champion for the cause of peasants and downtrodden. In fact, when he was a minister, he spent a substantial part of his salary for the scholarship and stipends for economically students coming from economically weaker background and bright students. So I'm really humbled and to accept this invitation to interact with you all through this webinar. So today I will be giving a talk on this nanomaterials for biological applications, a brief introduction, and then go for some of the work we have carried out in JNC. So I will be moving to the, I have to use the slides, some of the slides, which I need to go to the share things. I will move to the share document. Can I share the documents in this?
Yeah.
Yeah. Yeah. I hope you will be able to see this one.
Yeah, it is visual.
Yes, very good. So I am Ishwaramurthy, a professor in chemistry and physics of materials unit from Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, which is a small institute located at the outskirts of Bangalore, but now it is a part of the Bangalore. I do not know how many of you have visited this institute, but I would like to invite, if anyone comes to the Bangalore, please make a visit to our institute, and we are very happy to host you. This institute has some around 50 faculty members, people working in the areas of multidisciplinary, material science, biology, and theoretical science and engineering units. In fact, this center started 30 years back by none other than Bharat Ratna Professor C.N.R. Rao, and this vibrant research activities going on with around 300 research scholars doing PhD as well as master science. We are also having many programs for people who are doing MSc and BSc level in science career, so they can apply for summer research fellowship program, and then once selected, they can spend some time in this institute. So I'm happy to welcome all of you who are coming to the Bangalore. And we have many visitors, and it's a nice campus to look at it. Oh, it's not moving. Yeah. So we have the beautiful campus with a lot of greeneries and flowers, and you will definitely will enjoy. And we are getting visitors across the world throughout the year from national and international level. Occasionally, we are also getting visitors from natures who comes often with stay with us some time. So coming back, some of that I collected some pictures from about that Sranjeevi Chotu Ram name of this college. It's very interesting to read the history of Sir Chotu Ram, who as a one of the important personality during the independence era. So I'm very happy to be part of this webinar bearing the name of this legend. So coming back to this one, just to give a brief introduction about this nanomaterials. Probably you would have heard a lot about this topic, and still I would like to give a brief introduction on that for many students who are not familiar with this. So nanomaterials are the materials which are composed of nanoparticles or nanopores. You call it nanomaterials or nanostructured materials. Though they exhibit characteristic physical and chemical properties different from the bulk as a consequence of having at least one spatial dimension in the size range of 1 to 1000 nanometer. Time is not, yeah. So this is the classification which is given one of the book edited by Professor Tony Cheetham and Professor Rao, where they classified the nanomaterials in different ways, that nanocrystals, nanowires, nanotubes, nanoporous solid. Here the classification based on the size of the pores, which the pores are at the nanoscale, then you call it a nanoporous material. Even though the particle size may be bigger, they can be in micron, but the pores are in the nanometer range. You will call it a nanoporous solid. The famous examples are zeolites and aluminophosphates, etc. And if it is a thin film, it can be in 2D film, it can be 3D nanostructures. So various, depending upon the morphology and the particle size, you can classify in different ways. So they can be composed of metals, semiconductors, it can be carbon, it can be metal oxide, metal sulfide, anything which you want. The size and very important factor. So at the nanometer scale, properties of metal and semiconductors, they are in between bulk and atoms or molecules. They are size dependent. Surface atoms to bulk ratio is more, which I need not go back. Or just with one simple example here, you can see a ring, gold ring. This is not what you consider is a big ring to tell. So this has that one, for every one atom on the surface, there are 1 million atoms in the bulk. So if you look at that melting point of this ring, this is a bulk gold melting point, which is 1064 degrees centigrade. Now you take out a small sand size of gold, which is of 1 millimeter size. Now you look at that melting point, it is still 1064 degrees centigrade. Now you cut out even smaller, take out a size of 1 micrometer, which is the size of the red blood cells. Now look at that particles of 1 micrometer size melting point. You can still see that the melting point is 1064 degrees centigrade, which is similar to that bulk gold, which is of the ring size. So now you come down further, you take out a 10 nanometer and then look at the melting point. You will see that the melting point decreases very rapidly. And in fact, if a 1 nanometer size, you will see that it's almost like a liquid at 20 degrees centigrade. Even at room temperature, it becomes liquid like that. So that's a size decreases. It's a many properties, it changes, surface to bulk ratio increases when the size decreases. So it has a lot of applications. In fact, at that nanoscale, a metal can become a non-metal. Mercury, for example, 1 to 2 nanometer level shows a non-metallic band gap. So it is a mercury, we know the metal, but when the size decreases, it shows the band gap. So when the size goes down, you will see this electronic properties changing. So even inert gold, that's why we are wearing the gold items, where a lot of attraction for gold. So it is very inert. You can keep it hundreds of years. And even that gold become very active catalyst if you reduce the size to 2 nanometer. So it uses many of the photocatalytic applications. So even the color changes from the gold color to the red purple based on the size. So you would see that the plasma resonance gives you a lot of color, red or purple, etc. Even cadmium sulfide, bulk cadmium sulfide, which is 1700 degrees centigrade, if you reduce the size to 2 nanometer particles, then you'd see that its melting point goes to 800 degrees centigrade. So these are all that very important and interesting property change when you are going down to that nanometer size. And even this kind of famous Lycurgus curve, which is a 4th century, that Roman period, which is giving a red color, what you are seeing on the right side. I will take this pointer option. Yeah. So you can see this is the shine inside the cup. You will see that the transparent or transmittance light, you will see the red color. Whereas if you shine on the cup from outside, you will see the green color, which is coming from the scattering of this gold and the silver nanoparticles of 50 nanometer in size. So these are all that very interesting property. Even many of those Catholic churches in Europe, you would see the bright and beautiful colors coming out of this kind of nanoparticle scattering and plasma resonance. This is a very well known, you would have seen. In fact, this is a very interesting that the Maya blue here, if you place the dye, Maya blue dye, in the intercalories, so that nanocalories in the clay, so the lifetime of this dye increases, the photo bleaching will not be happening. So thereby these people are used extensively in those period to paint those kind of whatever statues. And even you can see those skulls which is having teeth which contains a lot of nanoparticles. If you look at it, our body, our skull or teeth, if you look at it and break it and looking at the microscope level, you will find there are very beautiful interplay between these nanoparticles of the calcium, the phosphate nanoparticles, and very nicely organized at that nanoscale, which gives the mechanical strength to the teeth and so on. So it's not that in use, so nature does very beautifully. It's a kind of biological engineering to say that. And you can see this leaves contains the nanoparticles of silica. There are certain North American country, there are leaves, some of the trees which contains nanoparticle of silica inside that leaves, and people use it as an abrasive to brush their teeth using these leaves. So it gives it a very good abrasive strength. And these particles also in different morphology, sheet-like, fibrillar, and globular, and even some of the sea creatures, limpets and chitins. These are the sea creatures which is very commonly found in water bodies, and they have that unusual tongue. What you are seeing is a tongue of a sea urchin. So, in fact, the Professor Lowenstrom, who is called the father of biometalysis. In fact, a separate area to discuss all those things, how the nature make use of these nanostructures for different purposes. In here, there is a tongue which is very strong. In fact, when he was sitting in one canal, Bahama Canal, he was observing some of these creatures goes and sticks to the rock. And when they are coming out of the rock, he found a lot of marks on the rock. They created a mark on the rock. So he was very curious to look at it. What makes them, these sea urchins, able to create that mark? So he took those species and took to that lab and examined the microscope, and he found that its tongue, which is made up of a lot of hooks, which further contains coated crystal iron hydroxide. These are all the small nano structure, nano rods embedded in the biomatrix, macromolecular matrix, which gives it very strong, very high strength. Even it can scratch the rock for their dining habit. It's a very brilliant composite to say that how the nanoparticle composites makes this, gives a lot of strength to that. And even the magnetotactic bacteria, which contains a lot of iron oxide nanoparticle Fe3O4 magnetic nanoparticles at the nanoscale, organized in one dimension and acting as a compass to navigate themselves in the north and south polar regions. So these are billion-year-old natural process and very effectively used by the creatures, animal as well as many water sea creatures. And people recently tried to understand that how to use this new properties things to that medical applications for the last few decades. In fact, they particularly, of course, they are using it for imaging and other applications, which I am not going to touch upon that. And mostly people trying to use it for drug delivery applications. Normally, when we are administering a drug, when we are taking a drug for some disease symptoms, they're all very untargeted, often toxic compounds act in a non-specific fashion. So they created that dose limiting complex effect, in fact, side effects very often. So this is where the nanotechnology, people are thinking it can offer a good solution, where by loading it with the nanoparticles, making it a nano dimension of drug, so they can enhance its solubility and its circulation in the blood will be a longer time. You can tune that way, and you can decrease its degradation and then concentrate the drug at the site of action. So these are all that new highlights where the nanotechnology can bring into this drug delivery. And in fact, why nanoparticle, if you see that a cancer patient with cisplatin and chemo agents, these are the medicines that they will be undertaking. This cisplatin action, we all know that they get into that nucleus of the cell and then get into the DNA, intercalate inside and then kill the cells. And they are completely non-specific. So this means this drug molecules and chemo, they can enter into a good cell as well as cancer cells. So that means you will have that post-chemo complications when they are taking it for a long period of time. The solution to the problem, you can encapsulate these drug molecules inside some kind of micelles and then administer it so they can get inside and release at a particular site of action, and thereby the possibility of going these drugs to that normal cell, healthy cell will be prevented. So that's that new approach, people trying to do that. In fact, there is some of that drug with this approach already in the market. For example, doxorubicin, one of the another drugs which used for cancer kind of disease, were now administered with putting the doxorubicin inside the liposome micelles, which is of 100 nanometer in size, and then delivered to that desired place. This is a new development which is already in the market. And the versatility of the nanomaterials is it's a beautiful surface chemistry. So the surface can be functionalized with a COH group. These are all thiol groups or charged species or amine groups and alcohol groups. So there is a variety of surface chemistry possible having these nanomaterials of 100 to 500 nanometer in size. And not only that, they can also have, you can make it in a variety of composition. You can completely make it in a complete organic, or you can go to that inorganic like a quantum dots, cadmium sulfide, and those kind of things. This can be different applications. So you have a variety of composition possible from organic to completely inorganic materials. And another possibility is that you can attach that many ligands, like small ligands or antibodies, even aptamers, peptides, proteins on the surface using the surface chemistry. So that is the targeting elements. By attaching antibodies, you can send it to that very site-specific uh, um, point, specific points where you want to release the drug. So that's the beauty of this, on the size in such a way that any of these molecules can be attached. Thereby, you can target the drug molecule at a particular place. And another thing is that you can also make these nanomaterials in a different forms. You can make it in a sphere, spherical, cubes, nanorods, and nanodiscs. So that is another beauty of this nanomaterials. So people make in a different way. In fact, if time permits, I may even give that how that morphology of this nanomaterials also influences the entry of nanoparticle into the cells. So there are decided characteristics for a nanoparticle to be used in the drug delivery. They should be inherently toxic. So your choice of materials are limited. You can't use the entire periodic table, the elements available. So it should be encapsulated, and they should be stable, colloidally stable in physiological conditions. They should circulate inside the body, biological fluid. And the clearance mechanism should be clear and should also circulate for longer time. So long clearance time is better. And they can be biologically or extrinsically controlled release a therapeutic agent. It should be possible. So these are all that very important, should be targeted. These are all the very characteristic features one should expect when you are dealing with nanoparticle for drug delivery. And one example here I would like to share. This one is a layered double hydroxide nanoclay, to say that. This nanoclay, which has a surface, is a kind of magnesium aluminum hydroxide layers. These are like a sheet-like structure. So this surface is a positively charged. So to compensate, there will be a lot of interlayer anions, carbonates, phosphate kind of anions. Normally when you prepare it, so you have that inorganic sheets intercalated with this kind of charge compensating anion. So these whole LDH, you can make it as a nanoscale in the sense of there are few nanometer in size. You can break into small piece and then remove this interlayer anions of carbonates and other phosphates when you are preparing it with DNA or ATP or even fluorescence agents. So people try to replace with the DNA and the ATP because these are all the negatively charged anions. So those carbonate anions or phosphate anions, which is intercalated during the preparation of that LDH material, layered double hydroxide material, can be replaced with this kind of DNA. So once you intercalate it, you will see that interlayer spacing, which is a very small here, it increases when you are intercalating with a larger molecule like a DNA. So once they are inside, they can be easily transferred into the cytoplasm of the cell, because normally if you have to send a DNA into the cell membranes are mostly negatively charged. So DNA, you know, it's a very negatively charged. So it cannot pass through this bilayer membrane of the cell, outer cell, because of charge repulsion, negatively charged repulsion. So, but when you intercalate the same biomolecule inside that layer, LDH layer, so now it's a charge neutral, which can easily pass through the cell, and where if it's a cancer cell and all this cytoplasm regime, the pH of the cell is little acidic in nature, around 5. At that pH, your layer, that's a magnesium aluminum hydroxide layer, the inorganic layer, which will decompose and leaving this DNA into the near the nucleus, which then get into the nucleus, and which shows that gene delivery applications. It's a very brilliant idea, very effectively demonstrated at the research level. And this is one of the things which the COI groups reported 10 years back. And we have used some of this clay, which is custom-made clay in our group, where this clay we call as a amino clay. This also contains inorganic layer. This is a magnesium phyllosilicate, which we call. This bar, what you are seeing, this is that inorganic magnesium silicate, which is a magnesium hydroxide brucide sandwiched between the two silica tetrahedra. This is a phyllosilicate we call. So this is linked with the pendants, the propylamine groups. Both the sides what you are seeing, these are the propylamine groups, covalently linked with the silicon groups in the silicon atoms in this inorganic sheets. So these propylamine groups, in fact, the pKa of the propylamine groups, they're around 8. So that means in the normal, if you make that clay, they're all stacked structure. If you put it in water, so the pKa of that amine groups are around 8. So water, the pH 7, the range, most of these amine groups are getting protonated. So once it's protonated, the charged repulsion makes this layer to exfoliate. So what you are seeing the precipitate in this vial, which is a white precipitate because of the white color because of that scattering of the larger particles, it's getting precipitated now. When you add water to this is in ethanol. When you add water to this, those amines group get protonated, then they exfoliates, and then you will see very transparent solution. In fact, here the layers are like more or less like nano layers. When you add ethanol, the charge are getting screened and then again undergo stacking, and you will end up with a bigger particles and forms a precipitate. So you can make a precipitate, adding water, you can go to that very clear solution, and then adding ethanol, you can again precipitate it. So this is a very simple way of dealing with this clay. No need to go for complicated procedures. Synthesis is also very simple, and we use it for many applications. So one applications here I would like to share this for the trapping the drug molecule. And the chemistry which we used is that this, I said, is amine groups, and we know that amine groups or aldehyde groups reacting with aldehyde groups reacting with amines, which is forming an imine. This is a reversible reaction based on the pH at which you are carrying out. At high pH, this condensation of that aldehyde and this amine takes place, leading to imine. This is at a pH of 8 above or above 7.5. You will see this right side product possible imine group. But if you reduce the pH less than 6, so this undergoes hydrolysis and gives back to the aldehyde and amine. So this is a very well-known reversible reaction, which amine interacting with aldehyde or ketone. So we use this simple chemistry. We know that the layers which contains amine groups, we brought in one aldehyde, that is a dialdehyde, which is having some alkyl chain, and we interacted with these amines, where that amine group of this layer and amine group of this bottom layer interact with aldehyde group of the dialdehyde groups of this molecule, and at high pH, these two are locked. So chemically linked, forming imine bond, and these two are locked. So when you carry out this reaction in presence of a drug molecule, you will see that many of the drug molecules which we have highlighted, this kind of the sphere, so they are all getting trapped because the two layers are chemically linked by this aldehyde and amine reaction forming the imine in between those drug molecules are trapped. So now you reduce the pH. This is at pH greater than 7, this reaction. When you reduce the pH, then you can see this can be released because this bond breaks, and thereby these two layers can be exfoliated in aqueous medium, and thereby the drug molecule can easily come out. So that is the idea. In fact, you can see that when the doxorubicin, the drug we used was a doxorubicin, which the color coming from those orange color is from this doxorubicin molecule in dissolved in water. It's highly soluble in water. And you can see the first one, that 1 hour we have kept it, this doxorubicin, in fact, at the 7.4 pH. Once we prepared this drug encapsulated or entrapped doxorubicin, this is the clay entrapped this doxorubicin molecule by doing this chemical reaction, bringing the dialdehyde with amine. So this is the one after the trapping you are seeing it. This is in solution or in aqueous medium at a pH 7.4. You would see that at pH 7.4. So what is at the bottom of this vial, which is having the color of this doxorubicin with the clay, all settle the bottom. So that means your doxorubicin is within the clay. It is not into the solution. And when the pH is reduced, you can see at 6.3, some of the doxorubicin molecules are coming out to that aqueous medium. And then at the pH 5.3 and 4.2, you see almost every doxorubicin in the solution. So this clearly shows that at pH above 7, the doxorubicin are very well trapped inside that clay galleries, and reduce the pH below 7, you'll see slowly the drug molecules are getting released because of the breakage of this imine bond. So this aminonucleate dibenzaldehyde without doxorubicin, also you can create gel at a pH where there is no color. You can see only when the doxorubicin is there, this color comes from that. Otherwise, clay dibenzaldehyde, whatever the compound we are taking, is a colorless one. And this clearly shows this diagram where this graph where the release property was measured with respect to time. So you can see that at pH 4.2 at pH 5.3, the black as well as the red dots, you can see within a few hours, we can say less than 1 hour, you will see the huge release of this drug molecule into the solution. Whereas the one which is kept at a pH 7.4 and pH 6.3, the green and blue, there is no release at all, even at 50 hours, more than 2 days or 3 days. So that clearly shows the simple chemistry, how you can trap if you can release this molecule at the site of action. So you'll be able to see the controlled release of those doxorubicin in the specific sites. And in fact, we have also used for a kind of what you call release of those some other drug molecules, which we have, some of the work is going on in our lab, which we will be able to publish in later stage, which I will be able to discuss in the next talk if I have a chance to discuss with you all. So in another approach, we have also used the nanoporous materials. These are all that materials which they have the pores, which is the nano regime, where 1 nanometer to something like a 10 nanometer, the pores are in the nano regime. These are the nanopores. And here very periodically organized pores also. So the pores, that is, it is made up of silica in this case. So these pores can be easily functionalized with the amine and other groups, which we used for different applications where we modify the pores from hydrophilic to hydrophobic in a reversible way. So we have used for another application here, where the pores are modified with amine group. The surface of the pores, the inner, whatever that the cylindrical pores, are modified with amine group as well as a phenyl boronic acid link. So you can have that surface propylamine groups, and then the propylamine attached with the amine group with the COH group form modified gives that phenyl boronic acid with the functional group diol with attached to the boron. So this is the picture which you would see inside the pore surface, where you have amine as well as the phenyl boronic acid modified structure, which is given here in this case. So what will happen at a pH 3? These amine groups are the low pH, so these amine groups are protonated. So that means these are all the neutral diol group, the boron diol groups, whereas the amines are positively charged. So that means the charge inside the pores at a lower pH is completely positively charged. When you go for higher pH at 8 and 9, you will see this amine groups, the protonated one gets deprotonated and becomes a neutral amine. So now the positive charge becomes a neutral on the amine group, and whereas the boron diol, this has become a borate anion at that higher pH. It forms a negative charge on its site. So the boron carries a negative charge, so forms a borate anion. At low pH, it's a neutral. So that means you will be able to see the charge reversal at pH 3, which was a positively charged pores, going to negatively charged pores at pH 9. So in between, you can see the both things, positive and negative. It's almost like a neutral surface to say. So this tuning, you can go from positively charged pores to negatively charged pores either by pH change, that means OH minus addition, or you can even create from positive charge to negative charge by adding glucose, because the glucose which strongly binds to that boron diol and forms that borate anion. So this is that important thing. So converts that boron diol to borate anion if the glucose is around. So you can convert that positive or neutral one surface to negatively charged surface if you have glucose, even at pH 7.4. So at pH 7.4, which is a neutral, negative, positive, but when you add glucose, it converts many of those boron diol groups into borate anion, makes that the whole surface can be negatively charged. So that means your charge reversal can happen not only by pH change, but also by adding glucose. So that is the idea which you wanted to use it to demonstrate. In fact, these are the zeta potential only to show that when the surface is only functionalized with the amine group, you will see that the surface charge, that's one of the way to measure that how positive the surface is, the zeta potential measurements. And where you can see it means from the 0 means it's a neutral. If it's a positive numbers, it gives that a positively charged surface. If a negative number gives a negatively charged surface. That's it, easy to view this graph. So you can see the surface is amine functionalized. At the low pH is a very highly positive charge, around 40 millivolts the potential. And you increase the pH, it goes down and almost comes to the neutral at pH 9, which you can see in this kind of case. If there is no phenyl boronic acid, you will see everything only amine groups. So here it's only amine groups, so you won't see any charge reversal. But if you bring in this kind of functional groups, then you will see when you are low pH, you are highly positively charged surface. Change to a high pH, you will see that charge was happening, which was at 0 at around 7.8 or 7 pH, and it increase the pH, the charge goes more and more negative. So complete charge reversal happening from positive charge to that negative charge. The same thing which you can even do the charge reversal by bringing the glucose. In this case, you can make it a positive charge surface to the negatively charged surface. So what we did with this, we introduced some of that dye molecule which to probe that whether this is really happening. That means what the idea was that when you are loading some drug which is in the positive charged pores, if the drug molecules are negatively charged, so you, through electrostatic interactions, you can load the drug molecule into the pores, positively charged pores. This negatively charged drug molecules can go and stay into the pores. Then you change the pH a higher pH. Now the charge, positive charge goes to negative charge. So that means what you are having your negatively charged drug molecules undergo repulsion, thereby they will be kicked out of that pores. So by charge repulsion, they can come out of that pores at a higher pH. At the lower pH, they were in attractive mode. At the higher pH, they are in diverse mode. So that was the idea. So we wanted to demonstrate using the dye molecule, which you can easily probe with the UV absorbance or fluorescence emission. And what we have done, this is a negatively charged dye, even at a pH 8 or 9, it's a negatively charged, it's because of sulfonate anion. And we loaded this dye molecule into the pores at a low pH at around 4, and then looked at variation of release with respect to pH. So you can see that percentage of the desorption of this dye molecule when you change the pH at different pH. In fact, we change the pH by adding a sodium hydroxide, very dilute sodium hydroxide. So start with the pH 4, there is no release. And then after 1 hour also no release. And then you increase the pH 4 to 6, there no release. Even another 1 hour, no release. Change it to pH 6.5, no release of the dye molecule. Because if it is releases, I would be able to see the fluorescence or absorbance of this dye molecule in the solution, the filtrate. And then you can see 7 slightly coming up, not much. pH 8, you will see that a sudden release within half an hour. You can see something 30% of release. And then pH 9, you will see the 80% release. So that's very clearly showing that our approach where the charged was able to kick out this negatively charged dye molecule from the pores. And even we have done the same thing with the glucose. With the same at the pH 7.4, we introduced the same dye molecule. And now we are changing the pH by adding the glucose. So that means the glucose will go under borane diol group where it will complex and forms a borate ester, which is negatively charged. So that means with the time, you can see that when there is no glucose, the release pattern is given here. There is not much release, even at 10 hours also. But when you are adding that 5 millimolar, you'll see that slowly the dye molecules are getting released. And if you are going for 100 millimolar, you'll see the sharp release even at 1 hour time. And then almost 50% of the dye molecules are released. In fact, that release also can be tuned by changing the concentration at different time interval. So you want to release at a particular time, wait for that and add certain amount of glucose. So here I want to release some 20%, add 5 millimolar of glucose, and then there is no release beyond that. And you want to further release, add a 25 millimolar, then you see another 20%. So you can stepwise release is possible at your will. So that's the idea, is that if we can trap that insulin in this strategy, will you be able to release the insulin or any of those molecules which will be useful for diabetic patient. So with respect to glucose concentration, whether this molecule can be released. That was the idea. In fact, we identified some complexes, which is in collaboration with some University of Trinidad and Tobago. There was a visitors who worked in our JNC in our group. So he come up with a complex which is known to kind of effective for diabetes kind of disease, where this complex can get into the cell and then initiates those kind of glucose uptake mechanism. So the problem is that we in fact, we trap this complex with the same, bringing the diol group into the system and put it in this borane diol kind of complex inside this one. When this forms a vanadiol complex bound to this borane diol. So when the glucose is present, so this will knock out this vanadiol complex, and thereby this can get into the cell and do the therapeutic action. So that was that idea which we have worked out. We have also found some positive results when we carry out the mice model diabetes-induced mice model, and hope that it will come out in a better way. Maybe in future we will be able to discuss on that. So this idea is that here you have to use this kind of glucose responsive delivery, the drug molecule, putting those molecules inside that nanopores. So that's that one way which we have carried out. Probably another 15, 20 minutes, I would like to go for another part where we have used the carbon nanostructure, carbon spheres for the delivery of drug molecule. In fact, an efficient drug delivery is as complex and expensive as the drug discovery itself. This is the biggest thing. Even you can find a drug very potential for the disease, but releasing at the particular point and targeted tissue is very challenging. So there are many macrophages, natural defense mechanism in our body will make sure that they cannot reach that point. So this one way we have to do some of those hiding the drug molecule and taking it and releasing some other places where we want. So one idea where we have used is the carbon nanospheres, which using this carbon sphere, we are able to deliver some small molecule inside the nucleus, which was these carbon spheres are derived from the glucose, which we can make it a size of 100 nanometer to 500 nanometer, which is of the scanning electron microscope, shows that the carbon sphere derived from hydrothermal synthesis of hydrothermal synthesis route using the glucose, saturated glucose solutions, gives that the spheres of almost uniform size, is around 300 to 400 nanometer. And these spheres are very interesting. They are amorphous in nature. They are very well dispersed in water, in ethanol or toluene. In fact, the dispersion in ethanol is very good. You can see here, even at 6 hours in ethanol, the dispersion is not settling, which indicates that the surface of that sphere may be more of amphiphilic in nature, which can be happy with both in water as well as in oil. So that's things. In fact, at the time when we are carrying out this study, there are many reports people use a carbon nanotube for drug delivery applications. So we thought of using these carbon spheres for the drug application. First test is to see whether these carbon spheres can get into the cells. And we had a very good collaboration with the molecular biology and genetics units, Professor Tapas Kundu, who is the director right now in Lucknow, what a Central Drug Research Institute, CDRI, Lucknow. So his group had a very good know-how about that biological studies. So we had a good collaborations. We are still having a few projects running together. So they were able to look at it, the molecule, whether the carbon spheres are getting into that cell by what are the standard mechanism, introducing these carbon spheres into the cell media. The HeLa cells are taken. The whatever you are seeing that the blue color, probably here this is not done. In the panel 3, you can see this is the nucleus of the cell, which is stained by heat stain. It gives a blue color in a fluorescence confocal imaging. And this is the cell membrane, outer cell membrane. You can see thin border, blue color line. So this is a cell nucleus, this is cell outer membrane. So you can see that when you put the carbon sphere into cell medium, and you are able to see in a 30 minutes itself that, yeah, the green fluorescence, what you are seeing, that light green fluorescence, which is coming from the carbon sphere. So the fluorescence are weak, nevertheless, you will be able to see this carbon spheres where it is going. So when you look at the cell, we will see in 30 minutes, the carbon spheres already inside the cells, crosses that outer membrane and gets into cytoplasm regime. So within 1 hour, you will see they are near the nucleus membrane, and in 3 hours, the panel is changed. So this A, B is 1 hour, and the C at 3 hours, you can see these carbon spheres, those green fluorescence coming from the carbon spheres already inside the nucleus, where the nucleus is stained with the histidine, which gives a blue color. And if you leave it at 24 hours, you see so many carbon spheres inside the nucleus. In fact, even the fluorescence imaging also gives the line imaging. You can see wherever the blue color comes from the nucleus, where there is no, without carbon sphere addition. After carbon sphere addition, you can see wherever the blue color comes, this emission from fluorescence emission, and you can see the green color, which is coming from the fluorescence emission from the carbon sphere. So that means you will see that the carbon spheres are well co-localized in the nucleus of the cell, which is very interesting for us. So we also did a cytotoxicity test, famous whatever the assays which biological people will be using it. And we found that the carbon sphere, the content which we are using is not at all cytotoxic. So that's a kind of standard test we have carried out to show that they are not toxic to the amount we are using. And then, okay, this we are able to see this carbon sphere able to enter into the cell nucleus itself. So then how to prove they are active? So this is where we had to take some molecule which is known to do some activation inside that nucleus. So we all know that, so the nucleus contains nucleo proteins, which has a lot DNA wrapped up over the histone proteins. It is a kind of what you call a chromatin structure, which contains histone proteins, histone, non-histone proteins, where you are seeing these white balls are the histone proteins. This rope-like structure is the DNA, so which has that kind of condensed structure as well as unwrapped structure. So this condensation, that is a wrapped or coiled structure, and unwrapping of this structure, these two are very dynamic in nature. It's called this condensed state, you can say close to chromatin. At this state, this is very inactive, and it is an open stage. Unwinding of those coil gives open structure. At this stage, it is very active, which is very essential. This state, going from this state to this state, is very essential, going from closed state to open state, because a lot of transcription, repair, and other mechanism based on how easily it can go to the coiled state to uncoiled state. So this is a very important step, dynamic in nature, and body controls and optimizes very brilliantly. And there are some enzymes which helps to do this process. In fact, you can see there are two enzymes, histone deacetylase enzymes and the histone acetyl transferase enzymes. In fact, this protein, the histone protein, has a lysine residue. So this contains amino groups. So this amino groups, when it is acetylated, the acetylation is done by histone acetylation protein, that is histone acetylation enzymes, that is histone acetyl transferase enzyme. So when it is acetylated, it weakens the hydrogen bonding interactions. So once acetylated, it goes from closed state to open state, because weakening the hydrogen bond interaction, so unwrapping happens, it goes to open state. So that is mostly controlled by the histone acetyl transferase enzyme. And from the open state to the closed state, you need to remove the acetylated part, the functional group. So that means you have to do the deacetylation, and thereby you get back to that lysine residue with the amine groups. So when the amine groups is that the hydrogen bonding is enriched, so thereby it can go back to the coiled state, where the closed state. So when it comes to the uncoiled state, histone acetyl transferase enzymes plays a major role, because it has to acetylate the amine groups and weaken the hydrogen bonding and open up the structure. And histone deacetylase enzyme does the reverse job. So remove the acetylated part, and thereby it goes to that lysine residue, and the hydrogen bonding interactions activated and goes to the condensed state. So this is a very important thing. So when you are acting HAT enzymes, it goes to the uncoiled state, thereby you undergo the transcriptional activation. When the HDAC enzyme acting on that, it goes to condensed state, there will be inactive. So these two are very well balanced. In fact, in the normal stage in the human being, you can see that in this both the acetylation process and deacetylation process by this histone HAT enzymes and the HDAC enzymes are very well balanced. But in abnormal conditions, for example, in this case, the HAT activation is reduced compared to the HDAC process. So that means the HAT activity, that the HAT enzymes is not very active to do the acetylation process. So this is imbalanced situation. So this kind of situation leads to that many complications, including those kind of cardiac, diabetes, neurodegenerative diseases, and cancer and genetic disorders. People have recorded, reported the kind of imbalance causing these kind of diseases. So one way to balance is you increase the HAT activity, and thereby this blue will be reaching that level of orange, and thereby you can come to that normal state. Or you can reduce that activity of that HDAC, you reduce this one, thereby these two can be equal. So one way to raise the blue or cut this red or the orange color. So normally in the kind of the drugs available in the market, there are that inhibits that this activity, HDAC enzymes activity, because when your acetylation level itself low, you don't want that deacetylation process to be very active. So rather you want to make the balance, you want to reduce this HDAC enzymes activity, thereby the de-escalation process will be controlled, and thereby whatever escalation are very well balanced. So that is the drug molecules people are having, molecules available in the market. They're all very non-specific and kind of creating a lot of side effects. One way is that the increasing the HAT activity, which is not much tried out in our kind of JNC, in our Professor Tapas Kundu's lab. They have come up with some, in fact, this HAT activity, the imbalance gives a lot of neurodegenerative diseases. In fact, they come up with this kind of cashew fruit derived molecule. This is called a CTPP molecule. This is extracted from the seeds of this fruit, and they modify a little bit and then come up with a small molecule which is called CTPP, which is known to activate the HAT enzyme. That means when you are able to activate the HAT enzymes, this can kickstart the acetylation process. So thereby you can see that activity of this blue bar increases. So that was that other approach which was successfully demonstrated in the Professor Kundu's lab. So the problem with this one, this molecule, if at all has to activate the acetylation, it has to get into the nucleus of the cell. The problem, this molecule is not able to get into that cell, which is the biggest problem. Unless it gets into the cell nucleus, its activity will not be shown. So this is where we realize that this carbon sphere, which can get into the nucleus, if these small molecules can be tacked into this carbon spheres, so they can carry the drug molecule into the cell and into the nucleus. So that's where the idea. So there it can be released, and then they can activate the HAT enzymes, and thereby increase that histone acetylation. So the process to study this, are many experiments which I am not going to discuss too much, only to say that how do you monitor that? In fact, when you have the carbon sphere attached with the CTP molecule, we know that it will enhance the acetylation of histone because it is activate the HAT enzymes. So HAT enzymes will further enhance acetylation of histone. So if you have any marker which can bind to that histone, that is anti-acetylated histone with the fluorescent tag, so they can bind to acetylated histone. So the Alexa 488 antibody, which can, that means the more acetylation of histone, the more Alexa, the fluorescent tag will bind to this one, and that means you will get a more fluorescence coming out of that. So this can be monitored by this way, how much acetylation happens when carbon sphere alone and CTPP alone, and carbon sphere and CTPP together, you pass it. Yes, how much time I'm having, sir?
Hello, Dr. Sudhir ji. Professor Siddharth Panda has joined.
Yeah. Hello.
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 So maybe
Dr.
another
Sudhir
5
ji,
minutes. How much time I'm having?
Professor Panda has joined.
Yes, sir. He has joined.
Yeah. So,
Sir,
5 minutes?
Professor Ishwar Murthy,
Yes.
sir,
Yeah.
please conclude in 2-3 minutes.
Fine.
Due to that, Dr. Panda has utilizing his lunch time for this talk. He is over busy today.
Yeah,
okay.
yeah. I'm just completing now. I just in
Okay,
3-4 minutes I will. Yeah, within.
Okay,
Yeah.
sir.
So you will be able to see that the kind of CSP CTPP carbon sphere loaded with the CTPP molecule, we are able to show that very good oscillation compared to the untreated without CTPP molecule, which we have demonstrated. So very interestingly, even these carbon spheres were able to cross the brain cells. That is one of the beauty of this carbon spheres we have identified. We use this property to deliver that some of the molecule. In fact, particularly drug molecules or anything to get into the brain cells, very, very difficult because the blood-brain barrier is one of the biggest barrier. No drug molecule can easily cross this barrier unless you do some tricks with that. Here we have prepared this carbon sphere from the glucose moieties. So this glucose mimicking surface functional groups helps this kind of nanoparticle carbon nanospheres get into that brain cell nucleus. Thereby we are able to show that by taking some of those interesting drug molecules, small molecule, you are able to show that demonstrate the development of neuron cells and the induces that memory in the mice model in a different way, which was in collaboration with a French group and which got published in Neuroscience a few years back. So these are all the various approaches we are able to use this carbon for some imaging purpose also, and even make these nanomaterials in different morphology, which I introduced in the beginning. So certain nanoparticles, if they are sphere or rod or kind of bowl shape, so depending upon the morphology, some gets into the nucleus, some gets into only cytoplasm regime. So we found that some of that iron oxide particle, which is coated with the carbon nanostructure, the glucose carbon. So one with the rod-like morphology, spindle-like morphology, or rod-like morphology goes only the cytoplasmic regime of the neuron cells, whereas that biconcave morphology gets into the nucleus. Thereby, we are able to deliver some drug molecule which is specific to the nucleus in this case, and there are some molecule which is specific to cytoplasm. So very important in future drug comes with that, how you are perfect in delivering the molecule at the specific site, even within the cell. Whether you want to deliver in a kind of cisplatin type of molecule, which for cancer active, disease, that those molecules should be delivered inside the nucleus, not in the cytoplasm. Whereas certain molecule, siRNA, those kind of things you may require to delivery in the HIV drugs, those kind of drugs to be delivered in the cytoplasm. So such kind of specificity very much essential for future success of this nanoparticle into the drug delivery, where we are able to show that some of that nanoparticles with that kind of iron oxide nanoparticle coated with this carbon, very nicely coated with the carbon, going into that specific sites and then showing the very site-specific action. So the drug delivered in that nucleus, you are showing very good histone oscillation for this TTK20, the drug molecule. When the Fe3O4 coated with the TTK21 with a biconcave, that is the morphology, gets into the nucleus, was able to show the immunohistone analysis. The dark stain comes from that delivery of those drug molecules inside the histone. The other one, weak stain, comes from the delivery of the same molecule in the cytoplasm. So you can see clearly one which is delivered inside the nucleus, one is delivered outside the nucleus. You can see clear-cut this activity difference in such cases. So with this, probably I would like to conclude my talk, where we are able to see how this kind of nanomaterials find a very interesting applications for drug delivery, whatever the important drugs into the nucleus of the cells, and particularly in the brain cells, where many of the neurodegenerative disease kind of problems can be addressed in future. So thank you once again for the organizers for giving the opportunity to give the talk here. And I'm very happy to discuss if any clarifications or questions are. Thank you so much.
Thanks a lot, Professor Murthy.
Yes.
We, along with our other participants, are enjoying your talk. You have started your talk from basic definition and classification of nanoparticle with examples, nicely explained how size change the property of the material, explain the nanomaterial for drug delivery, and how it is possible to utilize by increasing solubility and its surface chemistry of the nanomaterials and morphology of nanomaterial, effect of the how they affect the properties, and how can we incorporate the drug for human being and other living beings. But really, I have interrupted you due to that Dr. Siddharth Panda, Professor Siddharth Panda, has a over busy today. He accepted my this invitation last 29th September.
Yeah.
And
Sure.
utilizing
So I understand is scheduled, and the talk is 1 hour. I already finished what other
Thank
things
you,
I
sir.
have to tell.
Thank you, sir. Thank you, sir. Thank
So
you.
I would be happy in
nice
any of the, if anyone has a doubt, they can send me mail. I already shared my slides that address.
Okay.
So I'm happy to discuss anytime. So you can go ahead with that next talk. Thank you so much.
Once again, thank you so much, sir.
Thank you.
Nice
Thank
talk.
you.
Okay.
Bye.
Okay.
Yeah.
Now, I welcome to invite to Professor Siddharth Pandaji from IIT Kanpur for his talk. His talk title is Enhanced Performance in Ion Selective Field Effect Transistor. Dr. Siddharth Panda is currently a professor of chemical engineering and participating faculty in material science program, and the coordinator of the National Center for Flexible Electronics at IIT Kanpur. His research focus on chemical sensors for healthcare application and accompanying transport reaction transductions and material processing utilizing silicon and flexible printable platform. Prior to joining IIT Kanpur, Professor Panda was a staff advisor engineer at the IBM Semiconductor R&D Center, New York for over 6 years. He obtained a PhD from the University of Houston in 1999 and MS from University of Cincinnati in 95 and BTech from IIT Kharagpur in 1992, all in chemical engineering. Siddharth Panda has a credit of more than 150 papers of highly impacted journals. He has also a Golden Jubilee Chair Professor on Entrepreneurship and Innovation, Department of Chemical Engineering. Now I invite Professor Siddharth Pandaji for his talk.
Thank you, Professor Sharma, for this kind introduction.
Thank you.
And I would like to thank the organizers for this invitation and this opportunity to present our work. So can I go ahead and share the screen now?
Yes, sir, you can share.
Are you able to see it now, or? Just help me out.
Yeah, I think you can open your slides there. Yeah, we are able to see them.
Can you see this?
Yeah. You can go ahead.
Okay.
Yeah.
So I'm going to talk about enhanced performance in ion selective field effect transistors. And once again, thank you for this opportunity to present. So let me start with defining what are ion sensitive field effect transistors. So an ion sensitive field effect transistor is very similar to a field effect transistor in the sense that we have a semiconductor, we have a source, we have a drain, and then we have a insulator, and then we have a typical gate material in a typical field effect transistor. But here what we do is we replace the gate material with an ion selective interface. So when we have this analyte solution, typically an electrolyte, when it interacts with this ion selective interface, a potential is generated, and that is equivalent to what we have for the gate potential. Now this potential generated, that modulates the flow of charges between the source and the drain, and what we can have is now based on the current flow which we measured, we can now correlate a current flow with the voltage which is generated at this interface between the electrode and the electrolyte, and that which again is a function of the concentration. So thus we can measure the concentration of the analytes of this particular solution by looking at the characteristics of the transistors. Why we're doing this process? Because of the amplification capability of our transistors, and hence we can get very low limits of detections and very high sensitivities. So this gives a feel of what it really looks like. This is a solid state device, and these are the chips which are there. Now what we have is we have these applications in different areas. For example, we can have the medical field, for agriculture, environmental, and food processing, just to name a few. What I had mentioned here was like we have this electrolytes where the ions, and these ions again interact with the ion selective interface. Now these ions can be simple ions like this hydrogen ion, or they can be very complex ions like biomolecules. Now the mechanism of the interaction is different, whether it's for a simple molecule, for a simple ion versus a more complex ion. And in what I'm going to present is our work which spans all the way from simple molecules, the simple ions, all the way to complex ions. Now, I would like to give a comparison of a very conventional pH meter, a glass pH meter. I think almost all of us have seen or even used this kind of a pH meter. But what we have is when we talk of ISFET pH meters, they become solid state, and there are a few commercial companies which have these kind of solid state pH meters. Now, if you make a comparison between the this glass electrode based pH meters and the ISFET pH meter, this comparison can be done based on different parameters. But what I would like to say the difference is that while for glass electrode the typical size is about 100 millimeters square, for ISFET we only have a very small area. And again, if you look at the drift also is much less as far as ISFETs are concerned. While glass electrodes are more of individual types, these ISFETs can be integrated, and the advantage of that is that we can have large scale production compared to the individual production out here. Again, the times constants are much smaller, but there are several other challenges where, you know what, like this encapsulation becomes critical. So these are some of the advantages of this ISFETs. And now what we look at, like what are some of the performance parameters? We have seen some of the performance parameters out here, and one of them is sensitivity. So what we have done, we have been conducting research is how to improve this one performance parameter, which is the sensitivity. The sensitivity can be enhanced by several ways, but before that, let us look at the factors which affect the sensitivity. This is a schematic of a typical semiconductor insulator electrolyte system, and what we have is we are now looking at the different factors which affect the sensitivity. So first we look at the insulator. This is the insulator. Now we look at the different properties of the insulator, and also there's the interface of the insulator with the semiconductor also becomes critical. At the same time, we also look at the interface of the insulator with the electrolyte. So there are factors such as the type of surface sites as well as density of the surface sites that also plays a role as far as sensitivity is concerned. Now, what are the different means to enhance the sensitivity? There's several means which have been reported in the literature and which we also have looked into. One is utilizing high-K dielectrics and a stack. Another one is utilizing either nanostructure dielectrics or semiconductors. And the third one is using innovative device designs. Having seen these means to enhance sensitivity, let us make a brief literature review as to what some of the literature has to say about these different methods. One, if you look at use of different dielectric materials, there are some materials like silicon dioxide, which are very, very popularly used, and there's an ease of making them, but they have a relatively low sensitivity. On the other hand, we have got materials which are more exotic or more expensive, but they give higher sensitivity. So again, the challenge is, if you want to have a simple low-cost material, it gives low sensitivity. If you want to have a high-cost material, it gives high sensitivity. Another way is utilizing this nanostructured materials and dielectric materials. Again, there are reports in the literature where, for example, people have used silicon nanowires. In fact, they took silicon nanowires, they took a silicon surface, made nanowires on it, then deposited aluminum oxide. And then what was seen was that there was an increase of almost 10% from about 55 to about 60 millivolt per pH. Again, another method which has been shown is, again, zinc oxide nanowires have been used, and they have been capped with this amylopropane triethoxysilane. Again, that also has shown an improvement. The third method of improving the sensitivity is using a different architecture, and is a dual gate structure. And the first report on dual gate structure was published in 2012, where they went all the way from about 48 millivolt per pH to about 380 millivolt per pH. So that was a big difference, which was improvement, which was seen. So what I will do next is show some of our results, where again we have come up with different methods by which we enhance the sensitivity. The first one is utilizing nanostructures, that we utilize silicon dioxide nanoparticles, and also we chemically functionalize them to enhance sensitivity. So we utilized an electrolyte insulator semiconductor system, and this is a semiconductor, this is oxide. Then what we did was we put this nanoparticles on the oxide surface. After that, what was done was we put this we aminated it, and then this surface now was utilized for testing our sensitivity. So what we saw was that for a base case, it was about 37 millivolt per pH. When we textured it with different particle sizes, we saw an improvement. And what we saw that as the size of the particles went down, the sensitivity increased. So then what we saw here was that, like, by that there's an effect of the particle size on the enhancement of this sensitivity. So in order to do that, what we did, we had a more fundamental study that how to effectively texturize the surface. So what we used was we made an interplay of homogeneous and heterogeneous nucleation of the particles, and we came and what we found was there was a easy method to texturize these particles. That we dip this our wafers in a solution, in a sol of this growing solution, nucleating solution, this is a silicon dioxide particle, and by utilizing the appropriate conditions, we are able to get a good surface texture. Then what we saw was that if you look at this, we did the electrical studies on this characterization, and from this capacitance voltage measurements, what we found was that if you look at we measure the zeta potential, and for planar silicon dioxide, it was minus 36 millivolt. But when we modified with APTS, it came to plus 31 millivolt. And when it texturized it, it almost came to zero, in the sense it's almost like minus 9. So what this tells us that it is also, we're able to rationalize these results based on explanations, that when we have salinization, we have a certain protonation. And what happens is when we have the particle attachment, these NH3 plus sites get covered by the nanoparticles. So we have an explanation for for what we observed. Then what we did, we looked at the particular surface, and we found that when we have particles on the surface, there are enhanced defects, which this is what we saw from this UV absorption spectroscopy, and also from this photoluminescence results, that we see an increased defects when we have these particles, and the amount of the defects that increases with decrease in particle size. So that what we found is attributed to the reason for seeing higher sensitivity when we have texturing with particle sizes which are smaller. Then what we saw here was we looked at the device, and then again what we saw was based on this capacitor voltage measurements, we are able to characterize this device, which again explain the results what we see. For example, what we saw was this decreased capacitance after salinization, and that was explained by the lower dielectric constant of APTS. Again, we saw a negative flat band voltage shift, which is again attributed to the positive charging of the for the amine sites, and so on. So what was more important was that if you look at the positive flat band voltage shift, that was because of an interplay between this positive charge aminted sites and a negative charge on the silicon dioxide nanoparticles. So all what I would like to say is that there's a complex nature of the textured surface, which has this hydroxyl group as well as amine group, and based on the increased surface area, the particle size, number of defects, and all, we are able to get a high sensitivity. Now, having done that, initially we looked at utilizing silicon dioxide nanoparticles. Then what we wanted to study, could this same the findings work on a completely different system? So what we did, we again took nanostructures, but instead of nanoparticles, we took silicon dioxide nanoparticles, we took zinc nanorods. And then we again tried the same thing. And what we found, again, the sensitivity went up. So what you would like to say is that by utilizing this interplay between the chemical species and the nanostructured entities, we can improve the sensitivity, because you have seen that work with two different systems. What we did, this was initially we looked at this hydrogen ions, but then also we looked at complex biomolecules. So we tried this antigen, which is anti-PSA, and what we saw that we were able to get a label-free detection of PSA with the help of this kind of a system.
Professor Panda?
Yes.
I just want to, so you have carried out those sensitivity measurements at various pH, isn't it?
Yes.
And in the previous case, when you are moving with the amine functionalized, the APTS functionalized silica particles.
Yes.
So when you are going to higher pH, you may be having the charge imbalance because it becomes a more deprotonated form.
Correct.
So do you have any impact on that?
No, in fact, what happens is, if you look at the silicon oxide, typically they have this point of zero charges around pH of around 2.3, between 2 and 3. So what we have is, once we are typically, when we measuring from 4 to 9, because we are looking at physiological pH, we want to be around that. So typically all these things are typically negatively charged for the pH which we are using.
Yeah.
So what we do is, in fact, when we have our sensitivity, maybe I've not shown these curves here.
Okay.
What we do is, as a function of pH, we measure what is a shift in the threshold voltage, and by that process, this now becomes a pH meter.
Okay. And the kind of the stability of the high pH, the silica particles, do you have any comment on that?
Yes. So if you go, typically what many people have shown, they typically go between 2 to 12 pH. So what happens is not the silica particles, but when we go to pH of either 2 or 12, there are other factors, there are other films which get affected.
Okay.
So that is why we have kept around 4 to 9 is something which we have kept, and it has worked well. And what I will show in subsequent slides, that there are devices which we kept for more than a year, and we have done these stability studies.
Yeah.
I'll be presenting subsequently.
Yeah. So maybe your alkyl APTS functionality helps to stabilize a bit more, I think, because
yes. What we see is that that's a good interface between these two.
Yeah. So it could be more stable even at high pH also.
Yes.
I mean, functionalized. Yeah. Good. Thank you. Please go.
Okay. So then what we did, if you see here, we started with around 37 millivolt per pH. That's what we started. And what I will show is a journey that how we went on improving the sensitivity. And the sensitivity is measured in millivolt per pH. We started with around 37, and now we march along and how to improve the sensitivity value with every new concept. So then what we did next, we functionalized the this surface. And then what we did, we functionalized with branched polyethylene amine. Again, these are NH2 amine groups, but now we have a high density of these amine groups. So when we have a high density of the amine groups, we saw indeed that there was even a better performance. So what we have is, like, because of this, what we see here is because of the charging of this, the charging decharging of this amine group, we are able to see the accumulation and depletion. So this is a device, these are the capacitance voltage results. One with only the semiconductor, electrolyte and semiconductor with IXO. And the second one, the blue one, is when we have the ethylene amine. So now what we saw here was that we are able to get a much better performance out here. And what we see here is that, like, if we do not have that this BPA, we get about 24 millivolt per pH, but adding that BPA, we are able to get 110 millivolt per pH. So this is about 4.6 times the enhancement in the sensitivity. And another advantage is that, like, having this large density of this amine groups, we can now use it as a linker molecule for immobilized biomolecules. Then what we did was, we took this electrolyte gated FET and put it in a dual gate mode, that we changed the architecture. So in this particular case, what we did, we used a chemical coating and we saw an improvement. And then we took this concept further and added this dual gate architecture. Again, what we saw here was that we went from around 100, initially it was around 24, but now we are able to go to 160 millivolt per pH. So only by adding this chemical coating, we are able to go to 110. And by adding this new architecture, we are able to get to 160. So again, these are the things, this is the improvement what we saw. Then the question was that, okay, there's a lot of processing which goes into it, because it's not simply that, okay, we use a certain molecule and that enhances it, because for the functioning of a device, there's so many other factors which has to be taken into account. So now we have this indium gallium zinc oxide as a semiconductor. And then in order to have these molecules immobilized on surface, we also need to ensure that these surfaces are receptive. And also at the same time, these semiconductors also have to have a good condition for this for the mobility of the charges. So then what we saw here was, like, when we annealed it under different conditions, what we found that there was an optimum condition for annealing. So again, we have done detailed studies, and then what we found was that, like, at very high temperatures, there are bulk trap charges and interface states which are not needed. At low temperatures, obviously, it does not have the capability to perform well. So there's an optimum temperature, and this is what we found here. So then what we did, we again took the same device, but instead of reacting directly with the semiconductor, we added a dielectric. And in this particular case, we added tantalum oxide as a dielectric. And this is the structure of the device. Then what we did, what we found here, that when we annealed it under these conditions with oxygen at 400 degrees Celsius, that again gave the optimum conditions. Again, why? If we do not anneal it, there are oxygen vacancies at this interface between the oxide and the dielectric, and that has to be taken care of. So at 400 degrees Celsius for the optimum condition. Anything higher than that, again, we saw device degradation. So these are this XPA studies which we have done, and which shows why we are getting an optimum condition 400 degrees Celsius. And what was important here was, the pH has now gone to 400 millivolt per pH. So as you see, like earlier it was like 160 millivolt per pH now, but now we have been able to go to 400 millivolt per pH. And again, as a question was asked about stability. So what you see here, that these are the devices which we kept for 1.5 years as fabricated, then after 1.5 years, and after 1.5 years after clean and dried. So what you see here, that we took the device after 1.5 years, and again we clean and dried it. And what you can see here, that the device characteristics are the same or very close to the one which we even after 1.5 years. So this attests to the stability of our devices. Then what we did, we utilized a stack. As mentioned earlier, when we have a certain dielectric material, a dielectric has several properties. For example, requirements. For example, a dielectric should be, this should have high dielectric constant so that it can maximum capacitive coupling. It should have a high band gap so as to sustain the operating voltage. It should have low interface state so as to have better transistor characteristics. A high surface density of pH sensitive sites so that it can have a good buffering capacity. So these are all the requirements of the dielectric film, but no single dielectric can satisfy all the requirements. So if that's the case, what happens is that, like, depending on what we have, we may in one property it might be good, but other property might be bad. So what we did, in order to get the best of all, we decided to have these dielectrics, have a stack of dielectrics. Just to elaborate a little bit more, if you look at like the dielectric, like hafnium oxides, zirconia and all, they've got high dielectric constants, which is extremely helpful here. But they also have high inertian sensitivities, like this condition given, but they've got low band gaps. So that is why there are limitations for using this. On the other hand, you've got, if you have dielectric silicon dioxide, silicon nitride, and so on, they have got reasonably high band gaps, but they have got a low dielectric constant. So as I was mentioning, that no single dielectric can satisfy all the requirements. And so what we have done is we have utilized a stack of dielectrics so as to exploit the advantages of each particular layer. So then we made a device utilizing a stack, and this is the schematic which is shown here. So in this particular case, what we did, we took two dielectrics, we took yttrium oxide and tantalum oxide, and then we used different combinations of these, and we put it in this particular dual gate board. Then what we did, we underwent very rigorous study utilizing a MOS system. Then we looked at a TFT, we looked at dual gate TFT and dual gate SFET. So on the basis of this, all the devices tests which were done, what we found is that, like, I mean, these are all the detailed the results. But what I would like to say is that the presence of yttrium oxide and the interface of yttrium oxide with IGZO, that helped. That facilitated the performance of a device, and we got very good results. So all I would like to say is that when we have this different DGS FETs, we did this experimental, we did simulations, we also did theoretical, and from this we are able to have an understanding as to how the device performs. And what we saw here was that the role of this yttrium was very beneficial, and then this helped improving the interface with the semiconductor. And what we found was tantalum oxide, it interacted very well with the electrolyte, and the combination of yttrium oxide and tantalum oxide worked very well, and we got the highest sensitivities out here, which is around 450 plus millivolt per pH. Then what we did, we, instead of silicon dioxide for the bottom gate which we had, we now utilize the yttrium oxide combination. Now this also further improved the performance, and we went to almost 800 millivolt per pH. Then what we did, we utilized a completely different material. We used hafnium oxide. And again, what we did, I'm just going through the main results. We are able to get 900 millivolt per pH. But again, it's not simply just getting a material and using it. So many tests were done, and so many bugs, so many were taken care of before we are able to get to these numbers. Then what we did, so far we are working on this rigid substrates. Then we looked at, can we utilize flexible substrates? Because rigid substrates were again expensive. So in order to look at flexible substrates, what we did, we took a very simple material, which is this PET, simple plastic. Utilizing a very simple plastic, again, we went through these processes, but again, the processing is different because of the nature of the plastic itself. And then what we did, we saw that we again went down to 20 millivolt per pH, but that is what it is. As of now, the state of the art, as far as it's consists 37 millivolt per pH. For here, we reached about 20 millivolt per pH. Again, what we saw was like we bent it several times, and then again it was still, there was slight decrease in the performance, but never good enough. Again, we looked at the fabrication after 7 months. We looked at stability, a slight change in the performance, but again it still was working even after 7 months. Where will all these be used? Again, all this can be used in various things, like say packaging. It can be put in a package. It can be used for medical devices, and also it can also be used for wearable devices. We can just wrap it around the skin, and when we have got the fluid coming out from skin, we can be able to measure the pH. Then we also utilize the same system also for looking at our complex molecules. As I mentioned, here we are measuring hydrogen ions, and here we are using complex ions, which are the ions from the charged biomolecules. Again, now what we see here was, with the help of this particular device, we are able to go down to about 1 picogram per milliliter. That is the level of accuracy we were able, limit of detection we are able to go. Okay. Now, as I come to the end of the talk, I would like to again go back to where I started. That we are now measuring charged molecules. The hydrogen ion is the simplest ion, and the ion and this biomolecules are the more complex ions. Now, let us go back to a paper which was published about 15 years ago. And in that paper, what they have mentioned is that, like, what happens if you are measuring complex ions, which are complex molecules, versus simpler ions? What happens? What they have given, they give a very theoretical theoretical consideration, that if the pH sensitivity is high, then the sensitivity to complex molecules will be low. Hence, there's an inverse relationship. And then there was another experimental paper which came, which again their results did not match with this theoretical understanding. So then what we did, we wanted to understand this further, and we conducted very controlled tests. We took a device, and again what we did, we took a similar device. In one case, what we had was, we utilized it for measuring hydrogen ions. In another case, we utilized it to measure this complex biomolecules. And then what we saw here was, indeed, what we are saying, that wherever this pH, in our case, pH sensitivity was low, pH sensitivity was high, our sensitive biomolecules was low, and vice versa. So we want to understand, where is it, what is correct? Is the theoretical paper correct, or the experimental paper correct? Because they are giving two opposite reasons. So what we saw was, like, our results are matching with the theoretical paper, and we could see that the difference was that for experimental paper in literature, the way they had arranged the molecules were in a very random fashion. Our molecules were arranged in a more orderly fashion, and that is what we attribute the difference to. So what I would like to do is, right now I would like to summarize the talk, saying that the ion selective field effect transistors have advantages over conventional pH meters. And one of the performance parameters which we focused on was the sensitivity. And we looked at different methods to enhance the sensitivity. And these include using different dielectric stacks, using physical and chemical modification, different device designs. We also looked at flexible devices. The performance of today's flexible devices is very low compared to the conventional devices utilizing silicon. But again, this is a scope for improvement. And then also, we also address one important question in the literature. What, like for a given device, if the pH sensitivity is, if the hydrogen ion sensitivity is more, then the biomolecule sensitivity is less, and the vice versa. So with this, I would like to conclude the talk and acknowledge the efforts of all the students and postdoctoral fellows, all the collaborators and staff who have helped with this work, and the funding agencies, primarily the Department of Science and Technology, SCRB, and Ministry of Electronics and Information Technology. With this, I would like to thank you for your attention, and also would like to thank the organizer once again for this opportunity, and we'll be very happy to address any questions. Professor Sharma, you are on mute.
Thank you, Professor Siddharth Pandaji, for your nice talk. Nicely explained factors of effective sensitivity and how we improve the sensitivity, and explanation for the sensitivity of functionalization with, especially with pH, values. Thank you, sir.
Thank you very much.
Thank you. Thank you, sir.
Okay. Can I take leave now because there's other things to take care of?
Yeah. Okay.
And I wish you a very nice webinar. Thank you so much for organizing it.
Thank you, sir. Thank you, sir,
Okay.
for your
Bye.
kind
Bye.
acceptance. Now, our next speaker is Professor B.S. Keth from National Institute of Technology, Jalandhar. Now, I welcome and invite to Professor B.S. Keth Saab. Professor B.S. Keth joined NIT Jalandhar in 2007 as a professor of chemistry. Before joining NIT Jalandhar, he served NIT Hamirpur for about 16 years. He was head of chemistry department from September 2009 to 2012. He also served NIT Jalandhar in the capacity of Dean, Planning and Development, Registrar, Dean, Student Welfare and Dean Academics. Presently, he is head of the chemistry department. Professor Keth is HAG, higher administrative grade professor, which is the highest academic position in India. Presently, his research group is working on smart material and their application in sustained controlled drug delivery system, control release of agrochemicals, removal of toxic dyes and heavy metals, iron from the
Excuse me, sir.
wastewater.
Sarma sir, he is
Yeah.
not available right now. I think he is disconnected.
He is disconnected. He has joined.
He
I
has
have.
joined some, but right now he is not available. You can request him to join, please.
Yeah.
Yes, he has joined.
Hello, Dr. Sudhir ji. Hello, Dr. Sudhir ji.
Yes, आवाज आ रही, बोलिए सर.
Professor Keth Saab has joined.
Yes, sir, he has joined.
He
You
has
can
joined.
start your talk.
Yeah. Now, I welcome and invite to Professor B.S. Keth Saab for his talk at the topic functionalization of biomaterial and their application in different fields. Professor B.S. Keth Saab joined NIT Jalandhar in 2007 as a professor of chemistry. Before joining NIT Jalandhar, he served NIT Hamirpur for about 16 years. He has head of the chemistry department from September 2009 to 2012. He also served NIT Jalandhar in the capacity of Dean Planning and Development, Registrar, Dean Student Welfare and Dean Academics. Presently, he is head of the chemistry department. Professor Keth is HAG, higher administrative grade professor, which is highest academic position in India. Presently, his research group is working on smart material and their application in sustained controlled drug delivery system, control release of agrochemical, removal of toxic dyes and heavy metal ions from wastewater. He is also working on modification of fiber through graph copolymerization and nanogel composite. He has published more than 250 research papers in International Journal of reputed SCI and Scopus. He has produced 25 PhD students and 4 are going on. His citation index
Sir, ID Sharma sir, ID Sharma sir, you are muted.
कैसा? Hello? Hello? Ah, yeah.
Should I start?
Yeah, sir.
Okay, I'm going to my presentation now. Is it visual?
Yeah. No. Slide is not visual.
It's not visual?
No, sir, you didn't share your slides so far.
Okay, just a minute, please. Share screen. Is it visual? Hello?
Yes, sir, it
Yes,
is
sir.
visible.
It's visual. Okay, now I'm going to start my talk. Good afternoon, everybody. Before I start with my talk, I must thank the organizers of this webinar, especially Dr. ID Sharma, who invited me to deliver a talk in this webinar. And I'm highly thankful to him and other organizers of this college. My talk will be on functionalization of biomaterials and their applications in different fields. The contents of my talk will be brief introduction about hydrogels, self-healing hydrogels, introduction about nanogel composites, research work being carried out in our R&D laboratory, the need for wastewater management and work being carried out here at NIT Jalandhar, and eco-friendliness of hydrogels, because eco-friendliness is very important. Whatever material we are developing, whatever the devices we are developing, but they should be eco-friendly. They should not cause any harm to the environment. That's very important. And role of hydrogels in the retention of soil moisture and better growth of crops. That is the role of such materials in agriculture and horticulture sectors. So first of all, let's come to the brief introduction about the hydrogels. What are these hydrogels? They can be defined as three-dimensional hydrophilic polymeric networks capable of swelling in water or biological fluids and retaining fluid in swollen state. They can absorb 1,000 times of their dry weight. They can uptake the fluid to 1,000 times of their original weight. That's a property of such materials. That's why they behave like tissues also. They are three-dimensional polymeric networks and are highly insoluble in aqueous medium. They possess a lot of degree of flexibility and similar to natural tissues. They behave like natural human tissues. That's why they are used in the preparation of artificial organs also. They are of great importance in the preparation of artificial organs these days. Since these materials, they can respond to temperature, pH, light, magnetic field, electric field, ionic factors, and biological molecules, and we can call them as smart materials also, because they have the capacity to respond to the environmental stimuli. So we can call them as a smart materials also. And they are very smart. They can respond to pH of the surrounding medium. They can respond to the temperature of surrounding medium, ionic charge, and very important materials. And already told they are three-dimensional polymeric networks, and they consist of hydrophilic cross-linked macromolecules. And this three-dimensional network is responsible for absorbing and retaining the quantities of water or any other fluid. That's the beauty of such materials. Some of the characteristics of these materials are like high tensile strength. If you see the biomedical devices, if you want to use these materials for the preparation of artificial organs, definitely these materials should have good tensile strength, good compressive strength, and only then they can be used for the preparation of the artificial organs. Good thermodynamic compatibility must be there. And they must have the capacity to retain the structural integrity. That's very important. Like our cells. Our cells, if you press them, after some time, again, they come to the original shape. And these materials have that characteristic. That's why these materials are very important materials. And these materials have got a lot of chemical diversity. You can modify them as per your need. And chemical, they can be modified. And excellent biocompatibility. Yeah, this is a very important property of these materials. They have got least cytotoxicity, and because of the least toxicity or no cytotoxicity at all, they can be used in the preparation of biomedical devices. And they have got biocompatibility. They are compatible with our tissues. And that's why they can be used in biomedical fields. And high permeability and viscoelasticity properties also there. Say here, the smaller one is the before uptake of the liquid, whereas the larger figure, it is after the uptake of the liquid. Means you will see the structural integrity is maintained. There is no difference in structural integrity. That is maintained. That's the beauty of such our tissues. There's a capacity to maintain the structural integrity. You see here, you'll find that they can respond to pH, different types of pH. And if you see here, they show the pulsatile behavior here. If I take the material and I put that material at pH 8, it will swell. And after some time, I take it out and put it in another medium with pH, say 3, under acidic conditions. It will deswell. Again, I put the material in pH 8, it will swell. And you'll find it such a beautiful pulsatile behavior of such materials. That shows their smartness, how smartly these materials they behave. Swelling, deswelling, swelling, deswelling, depending upon the pH of the medium. So these are all the materials which we have prepared in our lab. These are our own materials. So next is the swelling studies in different salt solutions. You find that these materials, they respond very smartly toward the cationic charge. As you go on increasing the cationic charge of the salt, the swelling keeps on decreasing. Highly sensitive to the cationic charge. And at the same time, they are also highly sensitive towards the ionic concentration in the solution. As you keep on increasing the ionic concentration, the swelling behavior will keep on differing. So they are highly sensitive to the cationic charge as well as the concentration of salts in the aqueous medium. They behave very smartly. I will show you the results. Again, these are results of my own lab. And you'll find that with cationic charge, the percentage of swelling keeps on decreasing, and with engage in consumption of the salt solutions, you'll find that again the swelling of such materials will keep on decreasing. They behave very small it was the ionic charges as well as the concentration in the solution. So see the different applications of such materials in different fields. You'll find that they are used in wound dressings as well as to avoid any bacterial infection, microbial infection, and for the healing of the wounds. They are very important materials, and they are of great importance in transdermal drug delivery system. If you want to provide the medicine through the skin, they are great importance in that case. So transdermal drug delivery system. They are used very commonly in disposable diapers, especially for children and old people. And drug delivery, very important for sustained and controlled drug delivery systems. They are of great importance. And contact lenses, they are used in contact lenses these days, very commonly used. And tissue engineering, they are of great importance in tissue engineering these materials. Cosmetic industries, they are of great importance. Biosensor, these days they are used as biosensors also. And dye removals, toxic dye removals from the industrial effluents, they are of great importance. And in agriculture applications, you see, now this is the age of evergreen revolution. During 1967 to 78, 79, it was the age of green revolution. But because of green revolution, we are facing a lot of problems nowadays. A lot of toxicity in the food is there. In the food grains, fruits, vegetables, whatnot, in drinks, whatever you are eating or drinking, you are just eating and drinking the chemicals. I mean, that is because of the side effect of green revolution. Now, is this not the age of green revolution, age of evergreen revolution now? We have come to that, and in evergreen revolution, these materials are playing important role now. I will come to that. I will give a little bit details about that too. And in food packaging also, they are because of their better permeability for gases and exchange of gases. They are of great importance in food packaging also. Now, I will give you the little idea about how these materials are important in drug delivery systems. You see, when we go to the doctor, he will give you the tablet and your medicine, and he will ask you to take this medicine after every 6-hour interval. Why? Because the impact of any medicine, any drug in our body always depends upon the concentration of that medicine in the blood plasma. It always depends upon that. And you'll find in the figure, in the upper figure, you'll find that when you take the medicine, the concentration of that medicine in the blood plasma keeps on increasing, and it will reach the upper limit. And after, if it goes beyond that upper limit, definitely it will have toxic effects on our body, side effects on our body. And after some time, the concentration of that blood plasma keeps on decreasing and reaches the minimum level. And if the concentration of that medicine in the blood plasma is less than the minimum level, then it will have no impact at all. And then doctor will ask us to repeat this medicine after 6 hours. Again, we are taking the medicine, and the same graph you will find is repeated. Means definitely we are not getting the constant supply of the drug dose in our blood plasma. It is increasing when going beyond the toxic level, and then decreasing going beyond the limit. So minimum limit. So it means actually what we want, the drug supply, the supply of medicine to our body, it should be at a constant and optimum dose. That's very important here. But that's not the case in our traditional medicine system. It goes up and then comes down, goes up, then comes down. And that's why we are facing a lot of side effects because taking such allopathic medicines. These side effects are because of concentration of excess concentration of such medicine in our blood plasma. So under such circumstances, we should take the help of such smart materials. Before coming to that, let me again tell you, when we are taking the medicine, we are taking this medicine through if we are taking through the oral route, it goes to our stomach, and then the pH of our stomach is around 2. means it's highly acidic, hydrochloric acid is there. Under highly acidic conditions, that medicine may get decomposed. If it get metabolized here, definitely will have a lot of side effects. Even if it is not metabolized, anyhow, it will go to our intestine. In the intestine, the absorption of a medicine in the intestine will always depend upon the ionic character, chemical character of the medicine, ionic character of that medicine. Because through that, the intestinal tract, through the wall of the intestine, the medicine, the lipid layer is also there. Through that lipid layer, the medicine goes to the blood stream, and through blood stream goes to the desired place where it is required. But when we are talking about the ionic character of the medicine, what happens there? If your medicine is highly ionic in nature, it will be highly soluble in water. If it is polar in nature, it will be soluble in water. And you know, our body system contains around 70% water. Definitely, it will not get absorbed through that intestine wall to the bloodstream. Rather, it will be highly soluble in water. It will be flushed out of our body through the kidneys. Means that medicine will have no impact on our body. And if the medicine is non-polar in nature, then definitely what happens in that case? Here it will be the fat soluble, definitely. And you know, the intestinal wall, the wall of intestine, it has got liquid layer. Before going to the bloodstream, that medicine will get deposited over the lipid layer. Once that lipid layer gets saturated, then slowly the medicine gets diffused into the blood, and then definitely to the receptor site and all that. So what happens here actually? It always, the absorption of the medicine in our body and the reaching of medicine at the desired receptor site depends upon the ionic character of the body. That's why it is hardly 20 to 30% of the medicine which can be used by the body, rest goes waste. That's the main problem. I can give you here the examples of some of the such medicine. I will go in next slides, and then again I will come to this slide. See here, this is a gastrointestinal tract. This is the stomach, where pH is around 2. Highly acidic hydrochloric acid is there. You'll find it here. And you'll see the blood pH level of our body. The normal blood pH is around 7.35 to 7.45. You'll find here. And beyond that, and after that, it is all toxic in nature. So pH of our body plays an important role. आप बीमार होते हैं, स्वस्थ है, always depends upon the pH of your blood. That's very important. That always depends upon what we are eating and what are our food habits. It always depends upon that. See here, this is the different pH levels at different organs of our body. You'll find it a GI tract, how different pH are there. So I'm coming to the this dopamine. You saw this dopamine is called a deficiency of this dopamine causes Parkinson's disease, and it's very bad disease. And the dopamine, then you have to give dopamine from outside. Actually, it is given in the form of L-dopa. That's a product to dopamine. You cannot give dopamine directly. It's only L-dopa which is given. And because L-dopa can cross the blood-brain barriers, whereas dopamine cannot cross the blood-brain barrier. It always depends on the chemical structure of the medicine. So when this dopamine is given from our side, definitely this dopamine is traveling throughout your body. It's traveling throughout the body. And when it is traveling throughout, because it's required on the brain, but it has to travel throughout the body. If it is traveling throughout the body, definitely it is acting not as a medicine, it is acting as a hormone. It is not acting as a neurotransmitter here. It's acting as a hormone because it is traveling throughout the body. Then what happens when it's traveling throughout the body? It will switch on all the dopamine receptors of the body, all over the body. And if it's because you are not supposed to switch on all the dopamine receptors of our body. Only we require this dopamine in the brain. but it is switching on because it is traveling throughout our body. Definitely it will have a lot of side effects. That's the main problem in traditional medicine system. Next example I'm giving you here. Say, food-drug interaction. When you go to the doctor, definitely doctor will tell you, you eat this, after they're taking this medicine, you don't drink this, don't eat that, all that. Because doctor knows there's a food, and drug and food interaction is there, always there. Small example here I give you, see the tetracycline. What happens here? It has the capacity to bind with the calcium. Definitely, if it binds with the calcium, it will form a complex. And that coordination complex it is going to form, it cannot be easily absorbed in the body. Means taking this tetracycline, it's antibiotic in nature, carries no meaning if you are drinking milk after taking tetracycline. Because milk contains a lot of calcium. If you take calcium-rich food immediately after taking the tetracycline, that tetracycline will have no impact. Because of, and we should know that. Being scientists, we should know that what is going to happen. Drug-drug interaction. See, again, if sometimes we are taking 2 to 3 medicines together without knowing the scientific background of those medicines. And for example, cholestyramine is a cholesterol-lowering medicine. It lowers the cholesterol in the body. It is given by the doctors, those who have got high cholesterol level. But at the same time, if you take the warfarin and thyroxine, warfarin is again a blood thinning medicine. If you take this medicine with that, definitely they will form again a complex with each other. And those medicines will have no impact on the desired, against the desired the disease. So we should know what we should eat and what is the scientific background of such medicines and food and all that. Let's go to the anesthetics. Yes, you see, there are a lot of anesthetics in the market like desflurane, isoflurane, sevoflurane, and all that. These are the latest one. What happens? Because of their non-polar nature, they have the affinity for the lipid layer, fat layer. And in the intestine, there is a lot of lipid layer. First, because these anesthetics they are given if the doctor wants to operate the person. Before operation, the person is made unconscious through such anesthetics. But what happens actually? A lot of that such anesthetics they get saturated over the fat layer first before going to the bloodstream. Now, once that lipid layer it is saturated. And then slowly it goes to the blood and the person becomes unconscious and operation is there. Then afterwards, what happens? After the operation is over, so the patients come to the consciousness. होश आ गया उसको. लेकिन after some time, what happens? Again, the patients becomes unconscious. Why? Because a lot of such medicine they are deposited over the fat layer. They get slowly released into the blood. Again, after shocks are there, again he becomes unconscious, and that cycle is again and again repeated because of such problem. So let me give another very interesting example of tafenadine. It's a prodrug of fexofenadine. It's a fexofenadine. It's a prodrug of that, and this fexofenadine is the antihistaminic agent, and what happens? This fexofenadine cannot be given directly. It is given in the form of a prodrug. That this terfenadine, under the impact of enzymes, I have written here P450 enzymes. Under the impact of enzymatic reactions, this terfenadine it gets converted into the fexofenadine, which is the actual drug. But what actually what happens? If we after taking this terfenadine, this is a prodrug, if we drink grape juice, then what will happen? It will lower the enzymatic activity of the enzymes, that grape juice. And if it lowers the enzymatic activity of that those enzymes, that grape juice, the terfenadine will not be converted into the fexofenadine. Now this terphenadine will remain in the blood. And if this drug remains in the blood without getting converted into hexophenadine, definitely it will cause cardiac toxicity. Heart ka problem aana shuru ho jayega. That's the main problem. So we should never take the grape juice when we are taking the terphenadine. That's the main problem. So because that's how we should know the exact what we should eat and what we should not eat. These are the facts of phenadine actual drug. And see, now I'm coming to the previous slide now. I'm going back. Again, coming to that. Yes, that's right. Now, my point here was these materials, they play an important role because these materials are very smart materials. We can deliver the medicine at optimum concentration level, at a constant level, unloaded materials at the desired site. It will not have any side effects because it's not supposed to go through the whole of our body. The medicine is not going through the whole of the body. It's just we are giving just medicine just at the desired site only, especially in case of the cancer patients. That's why we are through traditional method. The cancer patient, they have to face a lot of side effects because the medicine is moved throughout the body, and the medicine is not in a position to recognize between the good and bad cells. It will damage all the cells, and people are facing a lot of side effects. So here, these some smart materials, because they are highly smart, they can respond to any stimuli. They can play an important role in such biomedical fields. One example I'm giving you here is the insulin, which is used for the control of the sugar in the blood. So definitely, mini pumps have come in the market, and which can be implanted, and they deliver the drug to the insulin in the blood, depending upon the concentration of sugar in the blood. So if the concentration of sugar is very high, it will immediately deliver insulin. If not, then it will not deliver. So these are the smart materials. Such materials can be used for such purposes. Then I'm coming to the other use of such materials, wound dressings. How important these materials are in wound dressings. They have the characteristic to minimize any scar formation. If you are delivering the medicine through such materials, there will not be any scar formation. They will minimize that. They strengthen the new tissues, and the strengthening of new tissues formation and provides protein for healing. They help in quick healing of the injuries and absorb fluids from the inflammation. They are again helping in quick healing of the injured place. They block the nerve endings to reduce the pain, encourages natural blood clottings, forms barrier against infection. They not allow the penetration of the microbial organs into the injured place, and provides scaffold for cell growth. They are very important. This factor is very important. Scaffold. They are used these days for the regeneration of, especially in case of your bones, for regeneration of bone cells. They act as scaffolds there, and they help in growth of the cells. They can be used as very good scaffolds there. Let me give you the example here. You'll find here, this is the hydrogel membrane made up of chitosan. Very important membrane. That's anti-hemorrhage in nature, and it's very important. Biomedically, it's very important membrane, consists of chitosan. And chitosan is available in India at the large concentration, no problem. So large quantity is available. It does not contain any therapeutic or pharmacological agent and does not interfere with the body's natural coagulation mechanism. It itself has got a medicinal value there, the membrane of chitosan. It is applied by simple pressure to the source of bleeding. If injury is there, you just apply this membrane at the source of bleeding and it will stop the bleeding. For a positive charge of this membrane sponge absorbs the blood and accelerates the aggregation of negatively charged platelets. Because the platelets in the blood, they are negatively charged, and definitely it will see the scientific background is there. Positively charged chitosan membrane and negatively charged platelets and aggregation of those platelets takes place. And acts as a mechanical barrier and prevents the blood loss, and by the accelerated formation of a temporary blood clot. This membrane, very important membrane in case of injuries, it helps in the temporary clotting of the blood whenever the injury is there. So this is I'm coming some a little bit chemistry also. See the kiting, that's a natural one, and the establishment takes place, and you'll get the chitosan, and you can. These are very important, biomedically they are highly biocompatible materials. Will list the cytotoxicity. So next, I'm coming to the next application of such materials. I'm coming to that. That is the self-healing. You know, self-healing is a natural process in our body. Automatically, our tissues, if damaged, they get, with the passage of time, healed up. That's a very important phenomena given by God in our body. Very important phenomena. So self-healing is one of the most fascinating features of biological systems, such as skin, bones, muscles, etc., etc. Very important feature, self-healing. So now the thing is, can we prepare such materials in the labs or not? Self-healing materials, can they be prepared and can be used for some biomedical importance or not? Yes, why not? We can. People are working in this direction. They are very important, very fascinating materials. Let us come to that. See here. Here you will find that constitutional dynamic chemistry plays an important role. They play a very important role. Constitutional dynamic chemistry is playing very important role here. You'll find you cut the material with knife into two parts, and after some time you will then put them together for some time. You find that they get joined up, they get healed up, and then you cannot separate them. That is the constitutional dynamic chemistry role here. See how important such materials are. They are very important materials from engineering point of view, from biomedical point of view also, and from other application point of view also. They are very important materials. They can be prepared in the lab. Here you'll find that dynamic association and dissociation of bonds takes place. Some of the bonds, they are initially broken, and some of the bonds, they are regenerated, and healing automatically takes place. So if you talk from engineering point of view, in mechanical engineering, they play an important role. In machines, if the formation of these micro cracks are there in machines, and they should get automatically healed up. In covering of machines, coatings of machines, coating should get automatically healed up, so that there is no damage to the surface of the machines. And if you talk about the biotechnology point of view, you'll find that they have great importance because such membranes can block the infection through microorganisms. Very important materials. You see here, these are two parts separate, and then when they were put together, and after some time, automatically they get healed up. Here, three parts, three separate parts. They were put together, and automatically they get healed up. Very important materials. So such smart materials which are having intrinsic ability of automatic damage, repair, or triggered by external stimuli. So they play an important role. Simultaneous formation of new bonds happen, and when old bonds are broken within the material itself. So that is a dynamic chemistry plays an important role here. Say how it happens. See, the self-healing materials are there, and with different types of bondings are taking place, are bonds. The covalent bondings and non-covalent bondings are there, and you'll find that automatically materials, they get healed up, self-healing. And such materials, they have a lot of applications, especially in cell delivery. When the injury is there, cells, they get damaged. So you can inject cells from outside through such materials. And you can inject drugs from outside through such materials without any further damage to the body. Very important materials. Tissue adhesiveness. They act as biosensors. They act as 3D printing materials. Very important. This is the latest these days. 3D printing in the biomedical fields. Very important materials. Antiseptic acids. Very important. And from engineering point of view, coatings as our sealants in automobiles, aerospace, building industry, etc., where self-filling is required. So again, I'm showing you the different types of bondings which are taking place in case of such materials. Non-covalent bonding, they they operate simultaneously, and the materials, in materials, self-filling takes place. See here, we have taken the animal who is injured, and we are injecting the drug from outside, and along with the gel. And self-healing of the tissues is taking place, and you will find both physical cross-linking and dynamic cross-linking is taking place simultaneously. And the fractured tissues, they get healed up with the passage of time. So that's the beauty of such materials. So this is the difference. You find at the extreme upper left-hand side, you find that drug delivery by intravenous injection and in situ drug delivery by self-filling hydrogels. In situ drug delivery by self-filling hydrogels is more advantageous than by the intravenous injections. They have a lot of advantage. You'll find yourself. Less toxicity, a better efficiency, and less frequency of injections are required. And that's the advantage of such materials over the intravenous injections. And other advantages are also there. Nano-based and nanomaterials based self-healing hydrogels. Yes, nowadays people are using nanomaterial based self-healing materials, like carbon-based materials are there, mineral-based materials are there, magnetic-based materials are there, and people are using such nanomaterials for this purpose. So I'm coming to the research work being carried out in our R&D laboratory at NIT Jalandhar. You will find that we have prepared some of the such self-healing material in the lab itself, and we have taken the biodegradable backbones, and gel formation could take place, and we could mold them into different shapes. And you'll find that a self-healing took place. These are in two colors. These are the results of our own lab itself. And even we have used such material for the antibacterial activity. And I've shown something here, and you find that they can block the bacterial penetration to the tissues. See, what kind of, this is our own work, work being carried out at NIT Jalandhar, and how the self-filling takes place, what chemistry is there, what type of linkages are there, you might linkages are there, and how the bondings are there. We have taken the CMC and borax and gelatin materials, and how the self-filling could take place. We could prepare very successfully such materials in the lab itself. These are some of the mechanisms. So these are all the results of our own lab. You'll find here how before self-healing and after self-healing and sealed materials, and these are all results of our own lab. And yes, you'll find the circular material is there, and disc is there, and we put that in the mid, and automatically after some time it got filled up. And these are the materials which we could prepare in our lab itself, and these are very good biocompatible materials that I will show you the results further. See, these are all the results of our own lab. How these materials in step 1, they are totally separate. In step 2, we brought them together, and in step 3, one of the side portion is got healed up, and you find ultimately a ring is formed, and it's a very good self-filling is there. Non-separable parts. So again, this is the work of our own lab itself. So you'll find yourself how the self-healing material can be prepared, and how they are healed up. These are all such materials. So then we studied the degradation behavior, because as I have already shown you, our body has got different pH at different levels. So it's very important to see to find out the degradation study of such materials at different pH. So we've studied all those studies. You carried out such studies at different pH levels, and you'll find that some of the materials they were quite stable at different pH up to 5 to 6 days, and those studies we could carry out. And we also carried out some rheological studies in the lab, and so as to see whether these are really biocompatible in nature or not, and they can be used in biomedical or not. And we also studied the hemolysis of such materials, and heme compatibility was carried out, and we found that the hemolysis ratio was found to be 2.73%, and if it is less than 5%, that shows that the material is biocompatible in nature, and we could find that. And we also prepared the nanogels of nanogel composites rather. So this is the definition of nanogels. I think I must not repeat all these nanogels and all that, because you have already overdose of nanomaterials, I think. So their applications. So we are now I'm again coming to the point here. Here we prepare some nanogel composites, and where we have used silver-nitride nanomaterials, and under the impact of gamma radiations, and we prepared IPNs and semi-IPNs. Here, semi-IPNs and IPNs were used for different studies. These are the radiation dose, gamma radiation dose, and you see that this is the physical appearance of such materials at variation in the physical appearance with radiation dose and with silver-nitride concentration. These are of IPN. They were of semi-IPN, and these are of that of IPN. So we also carried out some SAM studies here, and time studies also carried out. So these are of semi-IPNs. Time studies are carried out, and which will confirm the particle size. And we carried out the antibacterial properties of these materials, and we found that the nanogel composites rather containing silver-nitride particles, they were very effective against the both gram-positive and gram-negative bacteria. They were very effective against both the types of bacteria, and it was almost equal to those of the standard drug amoxicillin. Amoxicillin, again, which is the antibiotic, was taken as the standard drug here, and results were quite convincing. Here, these are the gram-positive and gram-negative bacteria, and the results of these nanogel composites. And this is the structure of the gram-positive bacteria and gram-negative bacteria, and you'll find here on the how the cell wall differs from that of the gram-negative bacteria. In case of gram-negative bacteria, it's very thin, and it's a peptoglycan cell wall is thick in case of gram-positive bacteria, and there's a difference between the two. And you find here what the mechanism involved, how the bacteria they get killed up with the help of such silver nanoparticles, and how the lysis of the bacterial cell wall takes place, and all the contents of the bacterial cell, they come out of the cell, and ultimately the bacteria lives. So we have this is the mechanism given by us, how these materials are effective against the bacterial infection, how they can be used in healing the injuries. So next, application of such materials in water treatment. Everyone knows that because we have to treat, and especially the industrial effluents, textile industrial effluents, drug industrial effluents, and other chemical influence. Chemical industry effluents, we have to treat those water because they are totally contaminated with different types of chemicals. And such materials play an important role in the treatment of such water waste. So, what type of work we are carrying out in our lab in this direction? You'll find here I will show you some of the results here. We have prepared a certain bismuth ferrite nanoparticles, and then with nano composites, we'll use them for the treatment of water. And we have prepared these nanogel composites with such nanoparticles, and we could use them very effectively against the treatment of water. And this is the characterization of such materials we carried out in SAM studies. And see here how these materials, they are very useful in degradation of dyes, mineralization of dyes. These materials are very useful, and they could degrade the dyes very effectively. And we have taken two dyes. One was cationic dye, other was anionic dye, and they could decompose those dyes. That is the beauty of such materials again. Say here, compared studies of the dye mineralization, uptake and mineralization. And again, this one, you will find that nano-composite gel was more effective than other samples. Again, we carried out the study, how many times such materials can be recycled, can be reused for this, for that purpose, for the uptake of dyes as well as their degradation, because degradation was also very important factor here. So we found that we could repeat it up to 5 cycles with almost a retention of efficiency. That is the both the cycles for both the dyes, Fe and CBB dyes, and they were very effective. Say here, this is how the dye degradation took place. These are all original results of our own lab, how the degradation and uptake of dye could take place. And we have studied all GCMS studies were also carried out for the uptake of dyes and their degradation for the fragmentation, and those fragmentations were further studied. Various fragments were obtained. Similarly, in other dye also, and all these studies were carried out. So these are compared studies of the demineralization of dyes in sunlight and UV radiation light. We carried out all those studies, and this is the values from the literature, and we compared our results with the literature value. Almost we found satisfactory results. These are our present study results. Yes. So further, as I already told you, because whatever of devices we are preparing in the lab, they must be eco-friendly in nature. They should not have any adverse impact on the nature. That's very important here. So we carried out these degradation studies, and degradation was studied under different conditions like soil burial method, composting method, and vermicomposting method. There are different conditions. We carried out such studies, and we found that these materials, they were biodegradable within 70 days. They're quite eco-friendly. That I'll show you how eco-friendly such materials are. So these are the all degradation studies we carried out under different conditions. These are all the results of our own lab. And the degradation of such materials were carried out at different stages: stage 1, stage 2, and final stage 3. And here we took the help of SAM studies and FTIR studies to see the impact of degradation on the materials. These are different SAM studies carried out at different degradation stages, and here the IR studies. And because you'll find here the functional groups kept on vanishing with the degradation as the degradation advances. So again, we also studied the soil where the degradation was carried out, of the degradation of sample carried out, whether there was any adverse impact of such materials on the fertility of the soil or not. That was very important part, because we should not poison the soil. Soil fertility should remain intact. And we carried out the macro analysis of the soil, where the samples were degraded. And we found that you see pH, organic carbon, available nitrogen, available phosphorus, available potassium, all those analyses were carried out, you'll find that the studies were carried out with respect to the control sample of the soil.
Hello,
And these are the samples. Yes.
Hello, Professor Kaisa. Sorry for interruption.
Yeah.
How long you are required? Are you free to conclude in 2-3 minutes?
2-3 minutes is too less. What happened?
Oh, last speaker is waiting so long. With you,
Any of that. Let me
conclude in
try.
5 minutes. Conclude in 5 minutes. Okay, sir.
Okay, let me try. Okay.
Okay.
Okay. So these we found that there was no impact on the soil fertility because soil fertility was retained here. That was the beauty of such materials here. And again, we found plant studies were carried out. They are using such soil containing the degraded samples, and you'll find that studies were carried out with controlled soil, and we got better results than the controlled soil. These are some of the results which we obtained, and some physical and parameters we measured there. And these are some of the studies which are carried out by your research group in the lab itself. And these are all the studies carried out in the lab and growth parameters taken. Yes, now we can say that such materials play an important role in different fields. Maybe you are biomedical, maybe you are engineering, maybe agrochemical sectors, maybe other sectors. Because these materials play an important role. And I would like to say here, we can contribute a lot, being a scientist, to the evergreen revolution, which is the need of the hour, because population is increasing at an alarming rate, and the soil is decreasing because of concrete. Jungle is coming up like anything, and expansion of buildings are there, and we have to think about that. Because we are consuming the fertile soil of the farms for the construction of different buildings, different types of buildings, and we have to think in direction. And such materials can play an important role in bringing out the evergreen revolution, because they are quite environment friendly. They can respond to different external stimuli. That's why we call them smart materials, and we can prepare the materials which are self-healing in nature, and they have great importance in both chemical in engineering point of view and from a medical point of view. Nanogel compounds can be used as antibacterial wound dressings also. That they are very effective also. They can also be utilized in industrial waste product management. Because of scarcity of time, I could not explain in full. They can help in removal of degradation of toxic dyes, and you can say they are quite eco-friendly in nature and can be used effectively as mini reservoirs. They can be used mini water reservoirs in the soil for the retention of the moisture in the soil for the better growth of the plants. They are of great importance. And our studies are going on in this direction in collaboration with University of Horticulture, University of Solon, and other industries, and better growth of crops during drought season, because this is very important. Food is more important for everybody. Technology comes later on. First is the food. So it's very important to see. Thank you. Jai Hind.
Thank you, sir.
Okay.
Thank you, Professor Kaisa, for your planned and nicely explanation for hydrogel and their application, highlighted their flexibility, characteristic of smart material, especially for the application of ophthalmologist, drug release system, and nanogel and self-healing.
Yeah.
Thank you so much.
Okay, welcome.
For your kind acceptance. And thank you for a nice talk.
Yeah, okay. Nice to meet you online.
Thank you, sir. Thank you, sir.
Okay, God bless you.
Good
Take
day,
care.
Good day.
Good day.
Now, I want to welcome and invite to Professor S.K. Tripathi ji from Punjab University, Chandigarh. Dr. S.K. Tripathi ji, presently Professor, Department of Physics, Punjab University, Chandigarh, and also coordinator at Center of Nanoscience and Nanotechnology, Punjab University, Chandigarh. His research experience research area, experimental condensed matter physics, nanoscience and nanotechnology, thin film and device fabrication. He has published more than 300 research papers, including review papers in the peer-reviewed and high-impacted journals, published more than 100 papers in international conference, national conference, symposium, and workshop. He edited number of books. He is member of different national body in country. He has supervised more than 40 students who has completed their PhD and more than 40 students for MPhil MTech projects. He had got fund for projects from DST, DRDO, CSIR and UGC. He has delivered many talks and chaired several sessions at different international and national conferences. Reference of many international journals. I invite to Professor S.K. Tripathi ji to deliver his talk. Professor S.K. Tripathi ji.
Hello, Dr. Sharma. Can you?
Yeah.
हाँ, मेरी आवाज आ रही है आपको?
Yeah, you are audible.
Okay, and video is also visible.
Yeah. Video is visible.
Okay. So okay, now
you may start.
Okay, now I will start. Good afternoon to all of you, and thanks to Dr. Sharma for giving me this opportunity to show some of my work, which we are doing at the Department of Physics, Punjab University, Chandigarh. And I think a lot of things may have been discussed about the nanomaterials. So I have chosen some different type of material, which is basically used for the non-linear device application point of view. Nowadays, the webinar or other things are only possible because of such type of materials which are used for the optical nonlinear devices and the communication purposes. So the title of my talk will be, that is, the role of radiation on the optical properties of glassy semiconductors for photonic applications. Before starting to this is my duty to tell you, to familiarize what type of work we are doing in our laboratory. Then I will come into our main topic. This Nirmal? It is not. So okay. So now I would like to tell you what are the work which we are doing in our laboratory. Basically, we have a very big group where more than 20 people in the form of PhD scholars, postdocs, MTech students, they are working in our laboratory. We have a very good preparation and characterization facilities in our laboratory. The materials which we are studying, these are basically the 2,6 nanomaterials, nanocomposites, maybe metal nanocomposites or semiconductor nanocomposites, 3,6 layered semiconductors, organic materials like rhodamine 6G, porphyrin, phthalocinin, etc. in the thin film form, especially for the gas sensing applications. Silicon and its alloys, silicon alloys, silicon carbide, silicon germanide, etc. Carbon and its alloys, maybe these are carbon nanotubes, graphene, reduced graphene oxide. Then oxide materials, these are basically the TiO2, zinc oxide, barium oxide, etc. Phase change materials, these are also known as the GST materials, that is basically the germanium antimonide telluride materials. Then magnetic materials and chalcogen-vitreous semiconductors, which today I will discuss with you. We have the different preparation facilities like chemical vapor deposition system, in which we have DC, RF and hot wire filament methods, blow discharge system we have, and different type of characterization facilities like transport. These may be thermoelectric properties or DC, AC conductivity, photoconductivity, Hall measurements, all those mobility measurements. Optical properties I will discuss later on. Structural properties and thermal properties like differential scanning calorimetry and all those things. Experiments I have already discussed some of them: DC, photo, AC conductivity, thermoelectric power measurements, space charge limited current measurements, time of flight experiment, constant photocurrent measurements, photothermal deflection spectroscopy, single slit photocarrier grating experiments, surface photovoltaic experiments, usually NIR, Z scan for the nonlinearity, photoluminescence, and differential scanning calorimetry. All these setups we have in our lab, and these materials are prepared and characterized for different applications like chemical or biological sensors, telecommunication, optoelectronic devices, PC RAM devices, and so on. In the last 5 years, we have published more than 60 international papers in the international journals, same in the conference symposium proceedings, and it edited 5 books. We have the funding from different agencies like DST, CSIR, UGC, NSC, DRDO, and all those things. We have different collaborations, national as well as international. These are TR Lab Canada, BARC, Mumbai, IIT Kanpur, IIT Delhi, IIT Madras, Nuclear Science Center, Delhi, Solid State Physics Laboratory, CSIO Chandigarh, and Semiconductor Complex Limited Chandigarh. So these are the collaborations which we have. Now I will discuss some of the things which we are doing in our laboratory. These are dye-sensitized metal oxide nanomaterials, for which we are using titanium dioxide and zinc oxide. Okay, I will not go into the details. And okay, so these materials are used for the dye-sensitized solar cells, LEDs, gas sensors, and water purifications, all those things. Sensitizing dyes which we are using, basically, these are metal-free organic dyes, inexpensive, which has large absorption coefficient and available in bulk with a cheaper rate. These are eosin, rose bengal, rhodamine B, ecridine orange, fast green, and all those things. These are some of the results, okay, like TEM image, transmission electron microscopy images, and from here we can see we have obtained the solid nano rods here, okay, and these from the XRD pictures you can see that at 450 and 600 degrees centigrade we have obtained the well crystalline NRTH structure in such type of structure. However, at 700 degrees centigrade we get the rutile phase of this much. Then we have used the dyes, and then we have obtained the absorbance spectra at different wavelength we have plotted here, and from here one can see that the dye-sensitized metal oxide films shows absorption bands in the visible region besides the UV visible absorption band due to oxides. After the dye is absorbed on the electrode, its absorption spectra broadened slightly, which we can see from the figure, okay, with the red shift in the peak position compared to the absorbance spectra in ethanol. Then electrical studies also we have seen, and then we have proposed a model you can see from here, and there you can see the mobility of the material, okay, after using the in the zinc oxide material, that is more as compared to the zinc oxide used in sensitized material. Then electrical parameters for zinc oxide, TiO2 and zinc oxide films here. And here we can see that the variation of photosensitivity is a function of temperature uh, and as a function of illumination intensity F and the time dependence of electrical conductivity during the rise and decay of all these three films in comparison. Okay, so then this we are also studying the thermoelectric materials, and here we are using the zinc oxide, bismuth telluride, cadmium telluride, antimony sulfide, antimony telluride, all these materials. And here I am just showing you the results of bismuth doped nanocrystalline zinc oxide thermoelectric materials. You can see from the TM image, which confirms the two component system with size around 45 nanometers or so. Spectroscopic analysis shows that the band gap of the obtained material is estimated using visual analysis, which is obtained to be 3.1 electron volt by the top plot, which is shown in the second figure. Okay, so thermal analysis also we have done, and from the TGA analysis, this gives the weight loss with respect to temperature, and from results we can conclude that the material has thermal stability till 950 degrees centigrade, and thereafter it decomposes. DSC gives the rate of weight loss, and from the graph we interpret that the reaction is exothermic in nature. Okay, these are some of the nanocomposite materials. Here I am showing you the lead selenide nanocomposite nanorods, and from here you can see that we have obtained the diameter of the nanorod is around 17 nanometer or so. Okay, this is for cadmium selenide nanocomposite, and here we can see we have obtained the bronze type of nanorods. However, in case of cadmium sulfide PV number nanocomposite, we are getting the spherical quantum dots also. Now we are also working on cadmium core cell structures. So there are two cases. In one case, the band gap is less of core cell as compared to the core. However, in the other case, the band gap is more in case of cell as compared to the core. Okay, so these are the two things, that is cadmium sulfide, cadmium selenide core cell, and the matrix which we have used polymer, that is polyvinyl alcohol also. So this is the XRD spectra of this much, and here we can see that in case of cadmium selenide PV nanocomposite, we have obtained the hexagonal structure, and peaks are occurring at 37.89 degree, 42.06 and 66.73, which comes from the attachment of silver to cadmium selenide, because we have added the silver in this particular composite. Silver has been added at different concentrations, and from here we have added different concentrations of silver, that is 10, 20 and 30 microliter, and from here we have obtained that the PL emission peak of cadmium selenide PVA is at 614 nanometer, which is due to the excitonic emission of cadmium selenide nanoparticles. This PL shifts towards the red spectrum due to the silver doping. So this is why it is so, because the quenching of PL intensity with silver doping is due to the efficient electron transfer from semiconductors to metal, which has been shown in the diagram here. Now these materials can be used for the memory devices. So we have fabricated the memory devices in the laboratory, and for that purpose we have used the two electrodes. One is silver and other one is the aluminum. Okay, and here you can see the current versus voltage for this memory device. And here we have seen that the memory device on-off ratio, which tells about the efficiency of any memory device. So here we can see when you add the silver with the cadmium selenide nanocomposite, the memory effect of the device that enhanced about 2.54 times. That is, it increases from 57 on-off ratio to 145 ratio or so. This is another picture. This is a core cell structure, and here we are showing the HRTM image of cadmium selenide cadmium sulfide core cell quantum dots. And here we are using cadmium selenide as 1.74 electron volts, which is a direct band gap, and cadmium sulfide 2.49. Okay, that is also direct band gap. And here from this HRTM picture, we can see that the image that the lattice fringes with spacing 3.5 angstroms are formed, which corresponds to the 200 plane of the hexagonal lattice. Okay, this one. Now, absorption and emission of cadmium selenide quantum dots. These are the actual pictures of the quantum dots which we have prepared in our laboratory, and you can see the different colors which we have obtained. Okay, so from here we can see from the PL spectrum that PL intensity of the cadmium selenide increased upon growth of cadmium selenide. The quantum yield increased from 10% to 19% upon the cell growth. Okay, this intensity is highest for the 7.6 pH environment, and agglomeration of the quantum dots is observed in the medium with pH lower than 6. Okay, so these are the observations. And these quantum dots we have used for the biomolecule conjugation, especially for the cancer cell treatment. One of the PhD students has studied this one. So here you can see we have used the trypsin drug, okay, that suggests the complex formation of cadmium selenide trypsin. Quenching rate, which we have obtained, that is 1.2 to 10 power 12. Binding constant this much, and binding site we have obtained. The binding of trypsin passivated the surface traits of quantum selenide, which enhance the fluorescent intensity. Now, fluorescence enhancement, which is taking place with the addition of trypsin, could be caused due to the determine the enzyme concentration which is present. We have also prepared the silver PVP, that is polyvinyl propyridine nanocomposite materials for the cancer cell treatment. And here we can see that as we increase the average molecular weight of PVP for this nanocomposite, we are getting the change in the shape from hexagonal to a needle type of needle-like structure. These nanoparticles, silver nanoparticles, these are used in various applications like photonics, optoelectronics, and biological labeling. You can see in figure C that the needle type of shape we have obtained when we increase the molecular weight of this PVP. Now here, these silver nanoparticles which we have prepared by green synthesis, that is, we are using the biological botanical items. For here we have used the ajwain, which all of us are taking in our daily life. Okay, so there we can see that we have obtained the surface plasma resonance band around 420 nanometer from the UV visual absorption spectra. Okay, and the TM structure, TM images shows that the dimensions of these nanoparticles ranges from 1.5 to 4 nanometer or so. These particles are spherical in shape with a smooth surface morphology. So these particles we have used for the cancer cell treatment. Raji cells we have taken. These were cultured with various concentrations of the above specified silver. For this, we have used the facilities of the biochemistry department of Punjab University, because this facility culturing we do not have in our lab. Okay, and this has been these synthesized green silver nanoparticles. These were checked for the cell viability. And using 20 microliter of this silver nanoparticle, this resulted in effective cell death, showing their toxic effect. The cell viability assay showed that smaller nanoparticles exert their toxic effect to a much larger extent as compared to the bigger nanoparticles. So these are the TM picture which we have prepared of the cadmium selenide thin films by glow discharge system method. And here you can see a very good spherical shape we have obtained of these cadmium selenide nanoparticles. And here we have changed the organ pressure inside the chamber during the preparation of these thin films. Different pressures, organ pressures we have used. and these pressures are 10 to the power -1, -2, and -5. And from this, one can see that the particle size, as we increase the pressure from -1, from -5 to -1, we are reducing the particle size from 20 nanometer to 5 nanometer or so. Then we have again prepared these cadmium doped with tin. Okay. Then again we have obtained the spherical nanoparticles, and the distribution shows that with increase in the amount of dopants, the particle size decreases. The shape prepared of these nanoparticles are spherical in shape, which I have shown uh, scanning electron microscopy pictures as well as the transmission electron microscopy images. Left side is TM images, right side is SEM images. Structure analysis of these have also been done, and it has been observed that as the amount of dopant increases, that is tin dopant increases, the crystalline character of the material decreases, and grain size also decreases from 19 nanometer to 13 nanometer or so. Okay. Then CPM result, constant photocurrent measurement results. Basically, constant photocurrent measurements, these are very, very important material for the thin film applications. You know, whenever you are preparing the films on some substrate, so a lot of dangling bonds and defects are created. Now the question is, how many defect density of states are present there? Because these number of defects, they will decide the application of these thin films for the optoelectronic applications or photonic applications, you can say. So from that point of view, from the device application point of view, you must know how many defect states are there, are present in that particular thin film or materials. So for that purpose, we are using the two techniques. One is constant photocurrent method, and the other one is photothermal deflection spectroscopy. These are the two techniques which are complementary to each other. By one method, that is with the help of photothermal deflection spectroscopy, you can get the surface defect states. However, with the help of constant photocurrent measurements, we can get the total number of defect states present in bulk as well as in the surface. Okay, so by subtracting uh, the results of CPM with the PDS, you can get separately also, that is the surface as well as the bulk density of defect states. So these measurements we are doing in our laboratory. The experiments have been designed, and we are doing, these are all computerized systems. So number of defect density and Urbach energy decreases with rise in annealing temperature that I have shown in these pictures. Now we come across our main topic. These are some of the things. We are also doing many other things, but due to the limitation of time, it is not possible to show you all of the results. Now we are coming our main topic. That is, the first question is, what are chalcogenide glasses? Every day in everyday life, all of us are using the glasses. These may be in the windows or the utensils or cup or something like else. But these glasses, which I will discuss today, these are slightly different than those glasses. Those glasses are known as the oxide glasses. They are insulators, okay, transparent, okay, and uh, semiconducting behavior they do not have. So they cannot be used for the semiconducting devices. Now what uh, BT columnists in USSR, they have basically replaced the oxygen which belongs to the sixth group of the periodic table. In place of oxygen, they are taking sulfur, selenium, and tellurium. Okay, so when they replace oxygen with any of these three or the combination of these three, then the glass which they have prepared, that is semiconducting in nature. Okay, and since these sulfur, selenium, and tellurium, these are known as the chalcogen elements, chemistry people are knowing to these. So that's why the glasses which are formed, those are known as the chalcogenide glasses. Okay, the glass. Okay, but BT columnists and his co-workers, they are not in a position to show any application of such type of material. So later on, after a few years or so, Osinski, yes, or Osinski, many of his people are knowing The name, he is a very well famous name, Xerox Corporation in USA. So S.R. Osinski, he has shown the first application in uh, uh, USA that these materials can be used for the memory devices, memory applications or switching applications have been shown by using these glasses. Since then, a lot of work has been started for the electro-optronic devices. But what will happen? Recently, from the last 10 to 15 years, their optical properties are very, very important due to the covalently bonded elements and which give them unique properties for infrared, nonlinear, and waveguide applications because these glasses they are transparent in the NIR or IR regions. So that's why for the waveguide applications and nonlinear applications, these materials are very useful. There are large values of nonlinearity at 1.55 micron, several orders of magnitude larger than the value for conventional silica glass that has been obtained. Okay, all of us are knowing that silica is used for the waveguide applications or optical fibers, but here the properties, the nonlinearity properties of chalcone glasses, that is more several orders of magnitudes. So that's why they are more important as compared to this one. Recently, third order optical nonlinearity of chalcone glasses has attracted considerable attention. So all of us are knowing the semi-empirical Miller's rule that third order optical nonlinear susceptibility chi3 that can be enhanced with the increase of the refractive index. All of us, most of us are knowing. Now, as the metal plays, metal ions which are inserting in these fast glasses, they play an important role in augmenting the value of refractive index. The addition of metal ions that may raise the value of refractive index and improve the third order optical nonlinear susceptibility chi3 comparing with the host materials. Okay. Now, these films which we are preparing, they have the low density of dangling bonds on the surface because of all the almost complete chemical bonds within the layer. Intercalation is there in case of films and mechanical weakness due to these weak van der Waals force between these layers. That's okay. Now it has also highly anisotropic transparent mechanical and optical properties and due to which high nonlinear optical coefficients in the infrared regions are present. And such type of properties are specialties making these materials a very good candidate for the second harmonic generation materials. So this is here I would like to show you here selenium, sulfur or tellurium. This belongs to the sixth group of the periodic table. And when you are adding the some metal, that will due to which what will happen? Conductivity increase in the crystalline structure. However, in the amorphous structure, the conductivity decreases. Okay. Similarly, activation energy is the reciprocal of the conductivity. So what will happen? Activation energy decreases in case of crystalline as well as increases in case of amorphous materials. Okay. All of us are knowing that the solids can be divided into three different categories. One is crystalline, other one is non-crystalline, and the third one is polycrystalline or so. So from here, we can see that energy gap in case of crystalline materials decreases and refractive index increases. However, in case of amorphous materials, van der Waals and covalent bonds, due to these properties, what will happen? The energy gap increase and refractive index decrease here. Now, due to which here we are taking the case of gallium selenide, suppose we are adding, and I will just show you some of the results of the gallium and then some later on some other results also. So gallium we are taking. So in case of gallium, basically this consists two gallium and two selenium sublayers in the sequence of selenium, gallium, gallium, selenium, where selenium, gallium and gallium, gallium bonds are covalent in the layers and selenium, selenium bond between adjacent four atomic layers which is shown in the figures here. You can see the green points, the, the, the broken bonds are known as the dangling bonds, okay? That is that. And van der Waals bonds is formed between the two similar atoms, between, or you can say that between the selenium atoms. However, that between the two dissimilar atoms, we are getting the covalent bonds, and also we are getting the covalent bonds between gallium-gallium metal atoms also. So how you can do, how we have done this much? We are taking basically the quartz tube where these materials which we want to prepare, they are weighed according to their atomic weight percentages and put inside the quartz ampoules, which is vacuum sealed here. Then we have put in the furnace where the rocking arrangement is there, and that rocking process is continuously taking place above the highest melting point of temperature of the individual materials for 24 hours or so. And then we get a solid when it is quenched in ice water or liquid nitrogen or so, we get the amorphous structure. Okay. Then using the thermal evaporation technique, with or without the presence of argon gas, we can deposit the films on the glass substrate. And here we are taking glass is the Corning 7059 glass at a vacuum of 2 into 10 to the power minus 5 millibar or so. Different gallium concentrations we have varied. Different, that is X is shown at 0.5, 0.5, 0.6, 0.7, and then we have deposited the thin films here. For this purpose, we have used this diffusion pump and coating unit. Diffusion pump is connected with the coating unit, and this is the actual film which we have deposited in the glass substrate. So you can see how uniform this film is, because uniformity is very, very important whenever you are dealing with the thin films. Otherwise, you will get a lot of defective states which are present in that particular material, which will not be a good material for the optical or photonic applications or so. This is a cryostat which we have used for the electrical measurements. This is, we have designed our laboratory for our specific purpose. Okay, and here you can see we have two glass windows, and through one side we are shining the light, and the other side we have put the UV visual photospectrophotometer. This system is attached with a liquid nitrogen trap, and all things are basically under the vacuum. And here the beauty of this system is you can measure the electrical as well as optical measurements simultaneously in the same system, because what will happen, most of the people, what they are doing the electrical measurements in one system and then taking the films and doing the optical measurements in other system, and they are comparing these results. So all the experimental conditions are different, so that is not a correct way of comparing the results each other. So it is a good thing we have thought, and we have then made design system where electrical as well as optical measurements can be done at different temperatures, from liquid nitrogen temperature to the above room temperature 400 Kelvin or so. Here some of the optical properties. This we are having a UV visual photospectrophotometer, Solar T2 2004, and this has the wavelength range from 400 to 1100 nanometers or so. Silicon photodetector we have used. And if you want to use further, that is up to 1700 nanometer or so, then we are using the indium gallium arsenide detector. The measurements have been done from 100 Kelvin to 400 Kelvin. We have used the Stanford method, for which we have developed a theory and simulation, and then the software has been developed, and these parameters have been calculated. All of us are knowing that optical absorption is controlled basically by the optical transitions, and you can see this is a top plot. Okay, so you can divide into three different regions, A, B, and C. A region is basically the transition between tail to tail state which is taking place. In the region B, tail and extended states, which follows, this is here the alpha follows the Urbach pool, and in region C you can see extended to extended states absorption is taking place, so which in this case alpha follows the Tox relation. And the fundamental absorption is that you can write alpha h nu equal to b h nu minus eg to the power n, the value of n that may have different values, half, 3 by 2 for direct allowed, and for forbidden transitions or indirect allowed and forbidden transitions, its value will be 2 and 3 and so on. So these are the formulae which I have used in the software. And here you can see the absorbance, that is x equal to exponential minus alpha d. I will not go into the detail because all of you are knowing. And from here we can calculate the refractive index in the weak and medium absorption region where alpha does not equal to 0 and the value of x is less than 1. So in that case, the value of refractive index n is given by this relation, and where capital N is given by this much. 2s, s is the refractive index of the substrate. T maxima, that is the maxima value of the transmittance, and T small m, that is the minima value of the transmittance. And from here one can calculate this much. So what will happen in the strong absorption region when it will take place? Fringes will disappear. You will not get any fringes, and so that in that case, n can be estimated by extrapolating it from other parts of the spectrum. Here you can calculate the absorbance using this different formulas. Absorption coefficient alpha equal to 4 pi n x upon d, and thickness of the film you can calculate by lambda 1 lambda 2 upon this denominator. Lambda 1 and lambda 2 are nothing. Basically, these are the wavelength of the two corresponding maxima or minima. From here, we can calculate the value of the optical gap, which can be determined from the intercept on the h nu axis between the plot alpha h nu to the power half versus h nu. And then different theories have been used for the determination of the oscillator strength, because all of us are knowing that these atoms are attached with a bond and these bonds are acting like a spring. Okay, so they have the bond strength, optical strength, and all those things which are represented by ed and e naught. So these values have been calculated from here. This is a relation y equal to mx plus c. So from the intercept and on the y-axis and slope, one can calculate the value of ed and e naught here. Now, temperature dependence of the energy gaps also we have done here. So different relations which have been used in case of semiconductors, that has been used, and from these relations we can also find out the steepness parameter, which is known as sigma, which basically characterizes the steepness of the straight line near the absorption edge, okay, which has been represented by this much. So again, the thermal variation of the band gap that is given is following by the following relation, which we are using here. Here the first term, that is Eg upon T, that represents, that depends on the lattice expansion, and the second term is basically depends on the electron phonon interaction. Okay. Then again we have used for the different formulas we have applied to see which theory fits in our experimental data. So for that purpose we have used the linear and nonlinear relation, that is Egt equal to Eg naught minus alpha t to the power p. Okay, and p is 1 for linear and p is equal to 2 for nonlinear. And then empirical law for Egt Watson equation, which we have used in this relation alpha beta, that depends upon the material properties. Third law, which is known as the Bose-Einstein equation. Here in this case, you can see that alpha b, that is a material parameter related to electron phonon interaction, which represents the strength of the electron phonon interaction, and theta b is the average effective phonon energy, which we have calculated. So these are some of the results which we have done in that particular sample holder from 100 Kelvin to 400 Kelvin. This is for the gallium selenide. The structure we have obtained is the amorphous in this case, and from these maxima and minima you can calculate the different parameters. From here we have plotted alpha h nu to the power half versus photon of energy, and from here we have calculated the band gap at different temperatures from 100 Kelvin to 400 Kelvins. These three samples we have taken: Ga2, gallium 2 selenide 3, gallium selenide 50-50, and then gallium 60% and selenium 40% or so. From here, then we have fitted these data, and then we have seen different theories we have put here linear, non-linear, Varsny and Bose-Einstein, which I have told you in the earlier. And from there, we have different parameters we have calculated for these materials, because these parameters are very, very important when you are going to design any optical device. These may be optical switches, optical amplifiers, or waveguides or so. So that's why these parameters are very, very important. Number one. And number two, suppose you are going to use such type of devices at a very low temperature, okay, maybe in the Siachen or where the temperature is very, very low. So you must know what are the properties of these materials, how the property or how the optical band gap or the linear chi3 or the value of the refractive index, how this will vary. Okay, so this you can see here. You know, class later. Okay. Then refractive index versus wavelength we have plotted here at 4 different temperatures: 100 Kelvin, 200 Kelvin, 300 Kelvin, and 400 Kelvin. And from here we have calculated the ED and E naught, and these values have been inserted in the table. We have calculated at all temperatures, but here we are putting just at 4 different temperatures: 100 Kelvin, 200 Kelvin, 300 Kelvin, and 400 Kelvin. Here you can, I have shown the value of E naught, that is oscillator energy, and ED, that is known as the oscillator strength. And from here you can see that the oscillator energy that decreases when you are increasing the concentration of gallium, and as well as the oscillator strength also. So now here we have plotted the energy gap at different concentration of gallium. 3 different concentrations we have taken, that is 40, 50, and 60. Okay, so here you can see that the temperature dependence of EG, that is fitted with 4 equations: linear, nonlinear, Varsny, and Bose-Einstein equations. Best fitting we have obtained with the Varsny equation. Here you can see the energy gap decreases with the increase in the metal percentage. Okay, when you are moving from 40% to 60% okay, of gallium, then energy gap decreases. I have already shown you in the graph. Now, increase in the energy gap we have obtained when you are decreasing the temperature. So this is the things which I would like to tell you here. Now, why decrease in the optical energy gap with increase in temperature? It is, you can see from this model which we have made here. Here the thermal expansion of the lattice coupled with the change of the electron energies with volume. What will happen when we increase the temperature? The band gap energy decreases because the crystal lattice expands and the interatomic bonds are weakened. Weaker bond means less energy is needed to break a bond and get an electron in the conduction band, which I have shown in the this model. Okay, and the other thing, number 2, contribution is the direct renormalization of band energies by electron-phonon interaction. With increase in temperature, the electron-phonon interaction increase, and this leads to reduced transport of electron from valence band to conduction band. Scattering, due to which what will happen? The transmission will decrease. Energy gap and refractive index also decrease with the increase in temperature due to this region. Okay. Then we have now we are irradiating these materials, because all of us are knowing that whenever you are using such type of devices for the photonic applications, different type of radiations in the environment that may take place. So for that purpose, we are doing the different irradiation treatments. For this purpose, we have varied the energy from milli-electron volt to mega electron volt. Okay, so that is done. Milli-electron volt energy that has been obtained with the annealing, that is heating it, keeping the material at some high temperature. Then plasma, argon ion plasma we have. Then laser, okay, light we have used, and then proton irradiation, that is 3 million electron volt energy. So we have taken the different energies to see which particular energy will be the most effective from here. And here we can see that at a particular wavelength, the refractive index decreases from 3.12 to 3.01 electron as we increase the energy. Similarly, for the energy gap also, we have done with the different same treatment. And here we have observed that uh, the energy gap we can increase from 1.45 electron volts to 1.85 electron volts or so. So then, because we have seen that the laser induced changes are getting the maximum changes, so we have studied in detail with the laser induced changes. This has been done at room temperature, and optical measurements parameters like refractive index, absorption coefficient, energy gap, K, epsilon 1, epsilon 2, these have been calculated before and after laser irradiations. For that purpose, we have used the green laser whose wavelength is 532 nanometer, and the different exposure times we have varied. That is 0 means without irradiation, then 500 second, 1500 second, and 3000 seconds. And then spectra has been plotted. Okay, and from here we can see that the transmission increases as the time of irradiation is increased, and these values have been calculated, which has been shown in these figures. Okay, so from here we can see here, that the value of refractive index decreases at the time of annealing. Time of irradiation is increases. Absorption coefficient, alpha is the absorption coefficient here, D is the thickness of the film, and X is the absorbance. So the value of K also decreases with the increase in the time of irradiation from 0 to 3000 seconds, and this follows the same trend as that of the refractive index. The real dielectric constant, epsilon 1 and epsilon 2, these are the real and imaginary dielectric constants. So the real dielectric constant epsilon 1, that can be calculated using the formula n square minus k square. However, the imaginary part, that can be calculated as epsilon 2 equal to 2 nk. The variation of real and imaginary parts, that has been shown in the figure here. And here you can see that these both parameters, that is the real and imaginary dielectric constant, they decreases with the increase in the time of irradiation. And the difference in the change in the energy gap, which we have calculated, that has been plotted at different exposure time. And from here you can see the difference increases as we increase the time of irradiation. Okay, then why it is so? Here we can see we have made the earlier diagram of the gallium selenium. So from there, if we take, we can say that the layer of gallium selenide consists of 2 gallium and 2 selenium sublayers in the sequence of selenium, gallium, gallium, selenium. These selenium, gallium and gallium, gallium bonds are covalent, and selenium, selenium bonds between adjacent 4 atomic layers is due to van der Waals force, which I have shown you by making a figure. Now these interlayer forces, that is selenium, selenium bonds, these are basically significantly weaker by a factor of around 50 than the strongest interlayer forces, which is between gallium, gallium bond. Light illumination, basically, that increases the randomness, okay, in the amorphous structure. So what will happen due to these photo-induced effects? That is consequences of some microstructural rearrangement which is taking place in the amorphous state, brought with the band gap light here. Photon irradiation induces large fluctuations in the selenium gallium bonds angle in a germanium selenium film. Okay, this reversible photostructural change due to increase in the volume of the film that is taking place. So the randomness in the structure increases after the laser irradiation. Now we have calculated the B half. Okay, B half is basically nothing that increases, that shows the disorderliness which is present in that particular material. So this B half increases from 378 to 428 centimeter inverse electron volt inverse. This indicates an increase in the disorder of gallium selenide and at different exposure times. The decrease in N refractive index and alpha suggests randomness in this gallium selenium increases. This I have shown to you. Now photosensitivity also decreases from 0.5 to 0.02 after irradiation, means it decreases almost 100 times, okay, in the defective state. So that also shows that this reduction in the photosensitivity, that is due to the increase in the number of defective states after the irradiation. Why it is so? Because light acts in this amorphous structure that weaken and break bonds of both sides, both types. Both types means van der Waal as well as covalent bonds. So these primary covalent bonds, known as intramolecular bonds also, and the weaker back bonds between non-bonded chalcogen atoms in the neighboring intermolecular bonds that is taking place. Okay? Gallium-gallium bonds act as structural defects and introduce localized electronic states into the lower part of the gap above the chalcogen lone pair p band at top of the valence band. Okay? If these gallium-gallium bonds is weakened, then they will go completely and they will form two neighboring gallium ganglion bond sites, due to which what will happen, gap state associated with the original gallium-gallium S bond orbital, that will disappear, and due to which it will give rise to two non-bonding gallium states lying near the conduction band edge. Okay, this raised above the level for an isolated atom by a p interaction with neighboring chalcogen in lone pair states. So net effect, what will happen, that net effect of bond breaking process will be gap states are replaced by states nearer the band edges, and the absorption edge in the gap region that will shift towards the higher energy, due to which photo bleaching effect will take place, and this will breaking of the existing heteropolar bonds and formation of new homopolar bonds will take place due to the absorption of high energy photon. So a change from gallium selenium to gallium gallium and selenium selenium bonds respectively, this explains the spatial expansion of the exposed films. This you can see so by the reaction, this equation also, that is this one. So through the absorption of light, chalcogen atom moves into radiation area from unradiated surrounding, and due to this motion of the of the movement of the atoms, chalcogen atoms, that will result a creation of new homopolar bonds in the irradiated area. Now, increase in homopolar bonds, that is gallium-gallium selenium density of defective states will increase, and the interlinking between bonds decrease due to the breaking of retropolar bonds, and due to which what will happen, absorption will decrease. Due to it, the density of material decreases, and therefore the value of N and K also decreases after the laser irradiation. Okay? Increase in density of the effective state, basically that means the concentration of free charge carriers decreases, and as we have seen that the photosensitivity increases from 0.5 to 0.02, it will confirm the increase in the density of defective states. Now I would like to calculate the third order optical nonlinear susceptibility in such type of materials. So before calculating this one, I would like to give you some basic theory which all of you have studied in the post-PG classes, that the linear optical susceptibility of isotropic medium, that is given by chi1 first order nonlinear optical, that is n square minus 1 upon 4 pi. And Wempel determinico single oscillator formula all of us are knowing, that is given by n square minus 1 E naught Ed upon E naught square minus Ch cos omega whole square, where E naught is nothing. I have already told you in the earlier that E naught is single oscillator energy and Ed is the dispersion energy, and H cross omega is the energy of light. So from the above equation, if you simplify it, one can get the first order optical nonlinearity chi1, that is given by ED naught upon 4 pi, this much. For high values of lambda, if the value of lambda is very high, in that case you can simply write it ED upon 4 pi E naught. This now the third optical third order nonlinear optical susceptibility chi3, that is given by the Miller's rule, especially for the visible and near infrared frequencies, and it is given by chi3 equal to AED naught upon 4 pi, this much, where n0 is nothing. Basically n0 is the linear index of refraction for h cross omega tends to 0. Now all of us are knowing that Miller has shown for a large number of materials that the value of constant A is approximately equal to 1.7 into 10 power minus 10. Okay, so the value of chi3 that you can write, if you put the value of A in the earlier equation, you can get chi3 is equal to 6.82 into 10 to the power minus 15 ED upon E naught to the power 4. So from here, we can see that we can calculate the value of chi3 using the values of ED and E naught, which we have calculated earlier by putting in that particular relation. Okay, so we can see here that the value of chi3 increases after the annealing as well as after the light radiation from these figures. You can see here the value of chi3 decreases as we increase the wavelength or decrease the energy. These are the four curves at different temperatures of this Ga2 gallium 40 and selenium 60 thin films at four different temperatures, 100, 200, 300 and 400 Kelvin also. This is this is for gallium 50 and selenium 50, and this is for antimony. We have checked in many other materials also. Antimony selenite, that is antimony 40%, selenium 60% at four different temperatures. Indium selenite, indium 40 and selenium 60 at four different temperatures. Then this is another one, indium 50, selenium 50. Okay, and then gallium, germanium 20, selenium 80, and germanium, selenium and silver doping with 10%. Okay, so and then annealing also of this germanium, selenium, silver, and then germanium, selenium, silver, light. So different materials we have taken just to see how the refractive index and the chi3 value of chi3 changes in all these cases. And from the comparison point of view, we have made in a single graph the value of chi3 has been plotted with respect to wavelength for these four different materials. One is germanium 20, selenium 80. Then germanium 20 and selenium 80, that has been added with 10%. And then annealing of germanium, selenium 10, and then light germanium, selenium and 10 light soaking has been done. And from here we can see that these results agrees well with the experimental values within a factor of 2 for many halides and sulfide materials. These nonlinear optical phenomena glasses are basically strongly related to some phenomena like nonlinear refractive index, multiphonon absorption, and stimulated Raman and Boolean scattering. This will also depend, the value of chi3 also depend the structure and composition of the glass, which are the major factors for controlling the nonlinearity of such type of materials. Here we have taken germanium 20, selenium 80, and when we add 10% silver on this, then the composition will change. Germanium 18, selenium 72, and silver 10 thin films. Here we can see that the selenium atoms are only two-fold coordinated and possess non-linear electrons, which are normally non-bonding. In chalcogenide-vitreous semiconductors, non-linear distortion of electron orbitals undergo light-induced reactions, which is easy to produce in such type of materials. Change in the local structure order in amorphous network occurs, which induces a change in the refractive index of that material. Correlation of nonlinear properties with composition are relatively well understood for binary as well as for binary chalcogenide-vitreous semiconductors. This lone pair approach, lone electron pair approach theory, that will give the nonlinearity of gases increases with the concentration of highly polarized one electron pairs. Then in this system, germanium selenium system, density of lone pair that will not change with the exposure, either with light or annealing which we are doing. Even then the refractive index changes. We have seen that the density of lone pair that does not change as we expose with the light or annealing. But even then we can see that the value of refractive index as well as the value of chi3 that change. Then the question is why it is so? So it is because the polarizability of electronic lone pairs of individual atoms is not the predominant factor influencing the nonlinear properties of germanium selenium system. Polarizability is not the only, which I would like to say, tell here. Some additional factors are also involved, which will influence the nonlinear properties. And these additional factors are glass structure, okay, density, presence of unpaired electrons, defect states. Those should also be taken care. In this particular material, germanium, selenium, silver, the structure of these glasses are basically the assembly of different structure units, GSE4 half and AG2SE. Now this AG2SE is basically they are present in the nanophase form in this system. Nanophase, okay? So situation is further complicated. Hello?
How much time required?
Just 2-3 minutes.
Okay, sir. Okay.
2-3 minutes.
Okay,
So
sir.
variety of different gaps that will also contribute to nonlinearity. So conclusion, what we can conclude here, we have prepared the gallium selenium, germanium selenium and germanium selenium system. Optical transmission measurement have been done at different temperatures. These parameters, optical parameters, have been calculated at different temperatures and band gap data, which have been fitted with different theories also. Annealing and exposed film with light of the germanium selenium system measurements like transmittation, optical parameters have been calculated. Chi3 value have been calculated at different wavelengths. This will increase as the temperature increased. This probably also increases with the annealing as well as light irradiation in germanium, selenium, silver. Probably lone pair orbitals density does not play the primary role in this increase. This also suggested that chalcogenide thin films should be exposed with light or annealed before their application. That will increase the value of Chi3, which are favorable or their potential use in telecom applications that might use highly nonlinear, high index contrast fiber or so. So thanks to all of you. I must give acknowledgement to my all such scholars because without that this work cannot be possible. So thanks to all of you.
Much informative for the research scholar field and for explanation for green synthesis of nanoparticle utilizing household commodity. That one is, for example, ajwain. Its important semiconductor used for solar cell, photo irradiation, explanation for change in photosensitivity with radiation. It is nicely explanation and much informative talk. Thank you, sir, for acceptance of our invitation at a short notice. Thank you, sir. Thank you. Now, over to Sri Bhupendra Singh for vote for next. Sri Bhupendra Singh.
Thank you, sir. Thank you, Sharma, sir. Am I audible? Hello?
Yeah.
Okay. Good afternoon. Okay, sir. Fine. Good afternoon to all. Honorable guest of honor, respected principal, sir, eminent speakers, and most valued participants. It gives me immense pleasure to propose vote of thanks on the behalf of Department of Physics, CCR PG College, Muzaffarnagar. I would like to extend special thanks to Honorable, our Chief Guest, our guest of honor, Professor Rajiv Kumar Gupta ji, who blessed us with his presence and took out a valuable time from his busy schedule. I especially thank to our eminent speakers, Professor M. Ishwarmurthy ji, Professor Siddharth Panda ji, Professor B.S. Keth ji, and Professor S.K. Tripathi ji for their valuable and innovative lecture on material science and nanotechnology. The way of explaining the topic was exemplary. I hope that definitely our students and younger researchers will be inspired and find a new direction in the field of science and research. A warm thanks goes to our principal, Sir Professor Naresh Malik ji, for inspiring, supporting, encouraging us to conduct and organizing such programs. Your dedication and commitment to our college always inspire us. I heartily grateful to all the participants for their active participation and listening patiently, as no program can be successful with a single person. So I extend my big thanks to my all colleagues and organizing team. I very especially thanks to Dr. ID Sharma ji, whose dedication has made this webinar wonderful. Thank you all for making this webinar